Cool. Okay. So yes, I'm Eric. I am the software architect at the Biomedical Engineering and Imaging Sciences Department of King's College London. I do a lot of software support, a lot of software help with people in the school in terms of their own individual projects and research. But I also am a major contributor to MONAI, and I've been there through the whole journey from the very beginning. So this is part of what I do as a software engineer working in research, as well as doing my own research. What I'm going to talk today about is some of the new things in MONAI, specifically on the bundle concept that we've introduced very recently, and a few of the other new ideas that we have associated with that, such as the zoo and some pathology and some other new features coming up in current versions of the package. So MONAI itself, of course, is a big library to help us do deep learning specifically with PyTorch. And so it's built on PyTorch to a huge degree. We are using a lot of the PyTorch ecosystem and building on top of that to provide some of the facilities that people need for doing specifically biomedical engineering AI research. So a lot of the data transforms and data loading, data handling, as well as networks and network components and loss functions and validation and some optimizers as well too. So there's a lot on top of that. One of the important things, obviously, with trying to have a deep learning model that is something that you can not just train, but then also use and distribute to other people, is having to have something that's going to have the information with it that is going to make that model actually usable. So with PyTorch, we can output a torch script model, and we know that that's going to be actually portable wherever we go, but it doesn't actually describe what it does, what the model is for, how to use the model, what the inputs are supposed to be, but the outputs are going to be, how to interpret the outputs and things like that. The idea with the bundle is to include all that semantic information into one package that you can then distribute to other people, and they can read it as humans, just reading the JSON files, for example. And these are meant to be human legible. And so they can look at this and be able to figure out what the model is for, what it does, what its inputs, outputs are, and so on and so forth. And you can also include with this any other information you like. So authorship, copyright, or license, or source of the data, or plain language descriptions. A lot of information you can put into the metadata.json file that comes in your bundle, which is just going to be a directory containing some subdirectories as shown here, with some config files and the actual model weights itself, as well as some README files and other documents. So the idea here is that we're going to include the saved weights for a model, as well as optionally the torch script model. You can also include the Onyx version of your model as well too. And so all that is going to be there that's compatible with PyTorch already. And it'll be the PyTorch objects that you're already familiar with, but we're layering on top of that in an additive way these configuration files that describe things in a more semantically rich manner. So you look at what's inside this bundle, and it's meant to be a self-contained package, but again, it's meant to be additive. This is a package that is going to be zipped into a zip file, or can actually be stored as extra files in a torch script object for that matter. But the idea here is we want to include the semantic information and also include workflows. So other JSON files that were there in the package describe specific workflows for training or for inference, and there'll be others for validation and for other application areas as well. The idea with these workflows is that the MONAI system can invoke these as if they were running programs and be able to construct a large set of objects that then represent your training environment, for example, or your inference environment. And so you can include with these JSON files a generic way of actually using this model and including the code to use this model in other contexts and for other users. Configuration provides, again, the readability and the usability for what these actual workflows are going to do, but separates them out from the Python code directly. These things are usable within a Python code. We'll see that later on. But the idea here is we're trying to separate out how we describe how to use the model from the actual code doing it, so that people can use their own code, or they don't have to have a specific software environment set up. We're trying to separate those concepts as much as we can, but still have a flexible way of defining a workflow and the components that are used in that workflow to use the model in a training or an inference environment. So there's other environments where we would use our bundles in, such as deploy, and we'll talk about that later on. So what we have in these JSON files are going to be definitions essentially of Python objects. So these configurations will allow us to create Python objects through the command line or in our actual code ourselves. And as I say, we'll see this later on. But this allows us to have some hybrid programming approach where we have configuration files that can be used to generate code, and we can do that in a lazy way whenever we want to, as well as ways of invoking them as actual full functional programs on the command line. We have ways of augmenting our JSON files with mechanisms for doing textual replacement or for doing object referencing. And so these are very powerful and important features that allows us to actually bind our objects together and allow us to actually override values that our objects rely upon on the command line or programmatically. We also maintain compatibility with TorchScript. So the idea with TorchScript, of course, is you can take a PyTorch network and package it up into a TorchScript model, which can then be used anyplace else without being tied to the actual software that was used to create that network. So this is a very powerful portable feature, and we just provide features on top of that. And so other software that uses TorchScript models can still use MONAI bundles. We're not tying you into our ecosystem in any hard way. I will include these slides later on, so you don't need to very quickly write down all these URLs, but these are the information and tutorial URLs that we point people to. So we have a number of tutorials in our tutorials repo for bundles and for other features. And we also have examples in the model zoo, which I'll talk about as well too. So looking briefly at what a configuration file looks like, this on the right, we have a configuration file for doing training of the spleen segmentation task from the decathlon data set. What you see here is the model definition. So we've abstractly defined what the arguments are to actually call the UNET constructor and create a UNET network object. And so we can separate out what those components are here into just what is essentially a JSON dictionary in this case. We also have at the bottom here some code that's representing the pre-processing pipeline. So this is something used to define a number of MONAI transforms. And as you see here, there's a few important keywords I'll go into in a little bit later. But in general, the idea to get from this is what the JSON looks like. We have a number of keys that are going to be defined in terms of textual values or in terms of Python code values or in terms of object references. And then these can be amalgamated together into a single workflow concept. And we'll see how to invoke that later. On the left, we have a number of examples of the actual command line invocation. I won't go into terribly much detail with these, but the idea here is that we can actually run one of these scripts as if it were a program. And we define on the command line what the actual components are we want to treat as being our program and what configuration files we want to use. And what the MONAI bundle library will do is be able to pull those config files in and load the appropriate objects to actually then run that workflow, whatever that is. So it could be inference, it could be a training run, it could be a validation run. We also have ways of validating the metadata of the network itself, as well as the bundle itself, as well as verifying the inputs and outputs of the network as defined by the bundle. So we have ways of actually validating that a bundle you've put together yourself is actually going to be a valid bundle and that it actually works. We do have also command lines for initializing a bundle, so that will create a lot of the template code for you, so you don't have to do a lot of writing your own boilerplate. And we have ways of exporting automatically to TorchScript. Looking more into the actual syntax of the configuration files themselves, this is an example of defining a transform for doing scaling of intensity values in an image. And this looks very much like how you'd actually create this object in Python. So the target special key here is something that refers to the class name, and this is what we're going to instantiate. And then the rest are going to be arguments for that class's constructor. So there's a very close analog between the JSON and the actual Python code itself. So here we're going to create this object. The keys argument is going to have just that value. Channel-wise will be set to true, and that will create that object. And that's something that will be created in the script at runtime or something you can do in code yourself. We could also instantiate other objects so long as we have those within our namespace. There's no problem doing that. So here we can create Torch's Atom Optimizer in the second one, and that works just fine. You might have to refer to objects by their fully qualified names, but it does work. Other little features as well too, where we can define whether an object is enabled or disabled based upon other conditions. That condition can be an actual Python expression that will be evaluated at instantiation time. And so this allows us to toggle certain components or features on and off depending upon the conditions that a script is being used in. We can also refer to other objects in scripts using the at operator, essentially. So in our JSON syntax, we are going to use at and hash and other characters as sigils, essentially. In this example here, data set is another object defined someplace else as a dictionary, and here we can refer to it when we're defining this object here. So of course, the chain of instantiation will have to be done in the background for you, but it can be done. And so this allows us to tie objects together. It makes sense. Obviously, data loader needs this data set, and this is the way that we go about doing that. We can also refer to members of objects in the very bottom here of our screen. We're referring to the train objects test LR member using the hash operator as essentially a member access operator. We also use $ to actually execute Python expressions. And so this is useful for doing evaluations to determine, for example, whether to define an object being enabled or not. It allows us also to pull up values that we actually want to use as arguments. So here we can actually, in the params value, use two of our sigils at once. So we're going to refer to the model object, but then actually execute calling as parameters method. And of course, this is the obvious and necessary way of setting up an optimizer. So that's what we have. We can also use the % operator to do actual macro replacement. So this will textually copy and replace values. And this makes sense in other contexts where you don't want to use reference using $ instead. So this allows us to bind objects together and build up a big structure that we would actually use for doing inference or for training. Looking at the command lines, again, the first one, this is how we'd actually invoke a training operation. We define what our metadata file, which is the top level information about what our bundle is, and then the actual configuration file we want to run. So that would be one of the training files, for example, for doing training. And the key here, training, refers to a series of expressions defined in that configuration file we want to actually run as the actual substance of our program. So those expressions will be executed one after another, and that defines what a program actually is. We can override values in our configuration file on the command line itself just by using $ and then the name of some value we want to change. And so this allows us to actually define different values for different runs, for example. So if we're doing training, we might want to change what a prefix is for where to store the weights so that they get stored in unique places for each run, or for each, or to change which fold we're using for the data set, and other configuration like that. So this allows us a lot of actual flexibility on the command line and not having to actually modify configuration files. We can also invoke these files and create the objects they define through code. So the config parser is the object used to actually do all of the actual operations defined in these configuration files. So it will read in the JSON files and then allow us to instantiate objects from them. And we can just refer to them by the names as defined in those JSON dictionaries. So here we can create the network def object, pull up as in channels. We can change what those values are and then actually create the object again to see what that does. And we call the method get parse content that will actually instantiate the object for us. And then that is going to be something that we can use right here and now in the code. And that object is going to be just a regular MONAI class. It's not going to be something special to bundles. It's going to be normal Python code, normal PyTorch objects, all the code we normally expect to see in as if we'd written it ourselves. But because it's going to be defined in configuration files, it's in a much more succinct and portable format for us. So tied very closely to our bundles is our model zoo. And this is a zoo that we're populating, of course, with exclusively bundles. So there's a number of examples we already have there in our zoo at this URL. And our zoo is just going to be this repository. The idea is that we're going to be accepting new bundles through pull requests, and then have our own validation mechanisms to ensure those bundles are valid and that they're versioned correctly. But it is going to be a public method of allowing new bundles to be stored in our zoo. So if you have something you'd like to submit, that this is the process to go about doing that. But the idea is that we're going to have just this repository for storing the bundles that we want to make public, and have our automatic tools for validating that these are correct, but also have automatic tools for downloading them as well. So the bundle library that comes with MONAI includes routines for downloading directly from the zoo by name. So the things that we store there, we can then refer to automatically and be able to then load and use dynamically at runtime without having to manually download things and unpackage them and figure out how to use them. So as I say, we're building a collection of segmentation models for deep edit and other MONAI label applications, pathology models, and there'll be others coming very soon. So just looking very briefly at what just the models looks like, it is just a normal repo on GitHub. So the models subdirectory contains all of our models, and then there's other information on how to submit a model, what the contributing guidelines are, and a lot of the continuous integration code that we have for validating models and making sure that they're all defined correctly. And in an example of a bundle that's stored here, you can see that there's actually very little in the repo itself. We have the configs directory that contains our configuration JSON files. The docs file subdirectory is going to contain our README file amongst other things such as the license. The large files.yaml file contains URLs to where the actual large files are stored. So these would be test data, the actual model weights, the torch script object, for example. So we're trying to not store large files here, and those are going to be stored in other places that our automatic download tools will know how to manage and be able to actually download data from. And see, we can have an example here of downloading this very model by version name, and this will automatically pull it down to a known local location, and then other software can use that bundle once it's downloaded. So these slides will be shared, as I say, so you don't have to worry about inspecting that too closely. So I'll talk very briefly on some of the new pathology features that we're adding to MONAI, but the next talk will go into more detail here. So we're adding new network definitions and new transforms and new facility classes to help workflows in the pathology realm that require very specific code to actually implement the kind of work that they need to do and to manipulate the kind of data that they typically work with. So for example, we have our WSI reader transform for reading this specialized file formats that a lot of pathology data is found in. Transformations for doing the sorts of manipulations that are a bit more specific to pathology. So for example, the transforms dealing with stain manipulation is there. When dealing with slide information, we often are doing things in a particular patch-wise way, and so we have a data set to help us deal with that kind of data and generate information from loading large amounts of data in a way that's efficient and effective for dealing with that sort of slide information. And we have applications that are being added to MONAI label for specific pathology applications. And we also are including enhancements to integrate with the Rapids QSIM library. And that is something, as I say, we'll go into more detail very shortly. More recently, and actually specifically the most recent release of MONAI, we introduced and have implemented a lot of concepts related to metatensor. So this is an idea that we're introducing into MONAI that is extending PyTorch's tensor class to include some metadata information with a tensor throughout its progress through transformation and network forward passing. the idea here is we want to include information about what the tensor actually is, what its actual semantic value is, such as its affine transformation, for example, if we're loading spatial data. We also want to include often with tensors what their history of transformation was. So what transforms were applied to that tensor, how can we use that information to implement the inverse transformation sequence to get that back to what its original state was. And that's something that makes sense for the inverse transform concepts that we have in MONAI. But all sorts of other information can be added to the metadata stored with these tensors. And this just makes a lot of our other transforms much simpler because that meta information is quite necessary. So when loading data from NIfty file, for example, the information stored in the header is needed by other transforms to transform that actual image tensor in a way that is a regular output. So making sure that the depth dimension is one axis or another, and because that varies depending on which NIfty, how the NIfty file was actually stored. So that information that is important semantic or structural information needs to be included with that tensor. And how we've done in the past was to include a separate dictionary with the PyTorch tensor object, but we're trying to combine those two concepts in an additive way, specifically in a way that should be compatible with all other PyTorch code. Our metatensor, as I said, inherits from tensor, and it should be compatible, it should be substitutable everywhere else where a PyTorch tensor is acceptable. But the idea here is we want to simplify the way we define our transforms and simplify the way that we can actually transport information along with our tensors that would be usable in other application areas. And that's something that we'll see over time as we change our existing transforms and adapt them to this new way of defining this metadata information. Very briefly, there's a number of other somewhat smaller features, but very important and very useful features added in the most recent release and in the most recent major release as well too. So the TCIA dataset object has been added for loading data from the cancer imaging archive. So this will automatically load the DICOM data and transform it into the 2D or 3D information, and with its image fully constructed together automatically for you. So you don't have to worry about manipulating the DICOM data directly. We're in the process of adding some more utilities for dealing with MRI reconstruction. So there's a subsection or apps directory for doing reconstruction and providing utility functions for doing that. And I believe there'll be tutorial examples as well. We are in the process of reviewing code for doing signal processing. So for dealing with one-dimensional signals such as ECG, EEG, and various transforms to augment that data. So doing sort of signal shifting or signal dropout, or doing other operations that make sense in the 1D realm that doesn't really have the same sort of meaning for images or for volumes. So that's being looked at right now, and we hopefully will have that integrated very soon. But it's quite an interesting area, and it'd be exciting to extend MONAI outside of the image domain, which is what we've entirely focused on so far right now. So stay tuned for that. We're also adding new networks all the time. We have UNETR and SWIN UNETR that are VIT-based UNet architectures that have been added recently, and these are based off of work from a lot of our MONAI our NVIDIA collaborators. So definitely check out those classes and their associated papers. HoverNet was added recently, as well as a network for doing segmentation classification in histology. So again, moving into other imaging areas away from just the CT and MR, which has primarily been our focus, certainly has been my focus as for research as well. We have many more small additions and optimizations, tweaks, and extra utilities added all the time. So definitely check out our releases, definitely check out our change logs, and see if there's anything really exciting that's come up for you. So that's it from me. And I guess any questions, and I'm not sure how we're going to handle it. We should be doing this all on Discord, I suppose. We can handle things as we go.
Yeah, I don't see any in Discord, but I do see one in Zoom. So for people who have questions, if you haven't joined Discord, let me go ahead and share actually really quickly. So people have the link again here. But we have our Discord server. You can go in there. There's a Q&A or question and answer channel on that left-hand sidebar that you'll find. And you can ask questions there as we sort of go through the rest of the meetup. Definitely ask them in there. I will ask the one that we had that was in Discord, and that was in Zoom. And I have a doc here that I'm sort of keeping track of all the different Q&A that I find as well. So one of the questions was, is it possible to add custom loss functions, transforms, models in a MONAI bundle?
Yeah, so I'm just gonna answer here just to mark it as answered, and then I'll answer verbally.
Yeah.
So MONAI is something that's additive on top of PyTorch. So we don't have a framework you have to buy into. If you have other code that's in PyTorch, it should be compatible with MONAI. I mean, there's nothing that we're adding here that you need to do. You don't have to inherit from any of our classes. You don't have to do sort of any kind of changes to your code or to or program to any sort of API other than PyTorch itself. So with MONAI and with bundles and with other concepts, everything is meant to be additive only. So if you have any PyTorch model already or other PyTorch code, that's something you can use with other MONAI code easily enough. You know, your loss function, your transforms, models, whatever, those can be used with MONAI. There's various ways of integrating existing transforms into our transform sequence, because we do have a few requirements there, but one way or another, we can get that to work. With bundles, it's the same story. If you have a model already, you have to fill in the metadata, that's true. But then if you already have the weights or the torch script model, you can just copy those files into the model's subdirectory of your bundle, and you should be off to the races.
Cool. I see some other here's one question that came in. How does bundles integrate with stream label deploy?
So that's something I think we'll be talking about later on. So I'm sort of giving away some of the surprises. With deploy, for example, we've recently put through our class to automatically load MONAI bundle and create an operator for using that. So within deploy, actual deploy applications are put together as a series of operators that are chained together. So an operator is something like a transform. Will take inputs and produce outputs, and that's how you produce an application in deploy as that sort of pipeline. And as we did in the past, you had to do things manually or adapt some existing code to get your network to work. But here, what we want to do is automatically generate an operator's internal structure from a bundle itself. So you should be able to automate a lot of the integration process to get your bundle into a deploy context. So we've approved that code just recently, and we have tutorials on how to use it, and we are now going to be destruction testing it to see how that works in practice. But I think we'll hear about that a bit later on. With stream and label, the process of getting bundles integrated into those libraries is currently ongoing, and we'll be, I think, tackling that in the next little while. But our objective is definitely to have those all seamlessly integrating with bundles, to be able to automatically go to bundle, and all the code that needs to be done to make that work in that particular context will be automatically generated from the metadata that the bundle includes. This is the whole idea, is we should have enough rich information in a bundle to be able to automatically do a lot of the stuff that you'd have to do manually, and eliminate boilerplate and other nastiness like that no one wants to deal with. So could you share more of your work on pathology, like the processes? So that would be the next talk.
Yeah. Yeah, so we'll get to that here up in just a second. I don't see, there's one more in
the Q&A section.
Zoom. Yes. Yeah. So is it possible to store information in bundles about the version of training data, indicating different strategies of pre-processing used to derive the model checkpoints?
Certainly you can do that. So in the actual bundle metadata, you can put any key you like in there. So you can put whatever you like. You can include multiple weights as well too, I suppose. As long as you name them separately, you can do that, and that's not a problem. So you can, for example, if you had a bundle and that was a network that was trained in a particular way, I would put the information about that into the metadata. And then if you had different slightly variations on that training strategy, you could include information in the metadata about how that was done, and then have different versions of the weights with separate names that indicate the relationship between those. Or you could create separate bundles where the actual metadata varies a little bit as you go. But putting everything into one single bundle is definitely possible to do that.
Cool. I don't see anything else for Q&A. I am going to go over one other section, Eric, before I wrap up with yours, which is, you already kind of touched on it already, but the model zoo. So I'll talk a little bit more about it again later on, but one of the things that we do have, and I'll link it in both Zoom and Discord, is that we have this model zoo survey. And the idea is, like Eric mentioned, we're trying to build that model zoo. If your team is, or your research is doing and using models already, are you using MONAI? Are you not using MONAI? Getting to know a little bit more about how you might be able to put your model into the model zoo. We might also be able to do a training session or walk through with you of how to package your particular model into a MONAI bundle and being able to get that published on the model zoo. So I'm going to put that here in general in Discord, and I'll go ahead and post it to everyone on Zoom. And I'll recap this again later on, but really we're trying to make sure that we get people, if you have models that you want to share, and you want to be someone who sort of has one of the first ones up on the model zoo and get that tutorial and instruction to help you get it there, fill out the survey and let us know what you're working on, and we'll have someone reach out to you. I don't know what the timeline on that is still completely, but I at least wanted to mention it now that Eric just finished talking about Model Zoo, and I'll talk about it again a little bit later today as well.
One question that came up on Discord, can I easily deploy the MONAI application on the phone? Unfortunately, we've done no work whatsoever on mobile. So if that's something that is going to be a large area of demand from the MONAI audience, that is something we could certainly start to get into, but which just hasn't been done yet.
Yeah. And one thing I do encourage people, if you do have use cases that you think would be interesting, that you want to maybe get a discussion going, go to GitHub. We have, and I'll link it as well in the different chats, but we have github.com/project-MONAI. The MONAI repo within there is MONAI core, but really whatever you're working on, if you want to know more about deploy and deployment, you'll have different sections for deploy, for label, for model zoo, whatever it is you're interested in, go in there and you can definitely start a discussion around what it is that you're interested in. If you have an idea, what you want to contribute, what's something you like, for instance, if a phone is something that you'd be interested and really want to get integrated, mention it in there, because that's really the big way that we'll know. The more people who can help start the discussion and contribute to that discussion, the more we'll know, hey, maybe this is something we should prioritize as something we want to integrate into MONAI. Cool. All right, awesome. Thank you so much, Eric. And I think you'll be sticking around a little bit. So if you have questions for Eric in Discord, feel free to message in either the Q&A. You can also reach out to people individually, but I encourage you, try to put it in Q&A or in general and talk about the things you're working on. And thank you again, Eric.
Okay, thank you.
So next up, we're actually going to talk about MONAI for pathology. So we had the Q&A question about pathology. Hopefully your questions get answered here. If not, and you have some more specific questions, we'll have another Q&A session at the end of this as well. But Shan, I think I put you on a panelist. You should be able to unmute yourself, and feel free to give yourself a better introduction. Go ahead and talk again really quickly.
Sorry, can you hear me?
There we go. That sounds better.
Okay, great. Thanks, Michael. Let me just put my screen. Okay, can you see my screen now?
We can see it.
Okay, great. Let me just put this. All right. Hi, everyone. I'm Shan Raza. I am assistant professor in computational pathology at Warwick. And I am part of the MONAI Pathology Development Working Group. So I'm going to talk about the developments in pathology in the MONAI framework today. So I know MONAI, Eric and Michael have discussed a lot about what MONAI is and some introduction to MONAI, but here I'm going to just focus on the bits which I will be discussing in the next few slides. So MONAI is open source Apache 2 license, which is very open license. And the main aim of the MONAI platform is to provide best practices for AI development in healthcare research. So here, most of the times, I mean, I've been working in this area of computational pathology for past 10 years, more than 10 years actually. But what we have seen is that the algorithms, especially in the area of deep learning, there have been many different algorithms developed, but there have been some issues with reproducibility of those algorithms when you take these algorithms from one center to another, and from after the publication of those algorithms, it becomes very difficult. So the main objectives here you have in the MONAI development we have been looking for is the code. The algorithms should be reproducible, they should be extensible, and they should be deployable as well. They should be easy integration as well of the algorithms developed. So I'll be discussing about that as well. And the code should be high grade here because when we are developing algorithms for healthcare, obviously there are strict regulations. If you have developed algorithm, it should be deployable in clinic as well. So that these are a few things which I wanted to mention before I move on. So the workflow, if I put it here, we have MONAI label, we have MONAI core, and then there is a MONAI deployment. So MONAI label is mainly a labeling and learning tool to create annotations. So when we are mainly working with pathology, we need a lot of annotations when we're working with pathology images and excerpts. So MONAI label provides us some tools for AI assisted to create AI-assisted annotations. This has been developed for obviously for radiology as well, CT, MRI, but I would be mainly discussing in my next slides on the pathology part, how it's integrated with existing tools. And MONAI core, and as Eric has mentioned in detail, it's a PyTorch-based framework. And it contains many different deep learning models. And then MONAI deploy is packaging, testing, deployment, and of these algorithms for clinical production. So Michael has already mentioned the GitHub page. If you want to have a look at what MONAI is, and if you want to work with if you want to learn about different algorithms and more about MONAI. Okay, so now just focusing on pathology, what I wanted to just give you guys a background on the kind of images we are working with in pathology. So this is an example of an image which we regularly work with on daily basis. The size of these images, these are gigapixel images. On average, the size of these images is 100,000 by 100,000 pixels. And we work at various levels in these images. We can work at the zoom out level, we can work at a low magnification, where we can look at the whole tissue region, or we can just zoom in all the way. For example, in this, as you can see, at 40x, where we are looking at the individual nuclei. There can be millions of nuclei within one tissue, and there are algorithms which can detect and classify these nuclei. And as a downstream analysis, we can perform various kinds of analysis, which involve, for example, spatial analysis, how these nuclei interact with each other locally, and they have implications on the clinical outcome of the patient. So we have millions of nuclei here. We can work at the region level, at the intermediate magnification levels, or we can just zoom out all the way to the tissue level and perform and develop algorithms at that level as well. So this is kind of a multi-resolution approach. So we need algorithms at each level if we consider this image as a pyramid. So this is what we are, the kind of images we work with, and obviously we need to develop when we develop algorithms for these kind of images, we need to have efficient pipelines, especially when we are looking at high zoom levels, because when we are at, for example, 40x, we need to process the whole slide image, this whole image, and it can take quite a lot of time if the algorithms are not efficient. And GPUs, of course, and they help us to make these process these images efficiently. So when we're talking about optimized for pathology, again, the algorithms, so the code should be customizable, and it should be available for users who have varying levels of expertise. These algorithms should be, the code should be easily integrated into existing workflows. And there are some domain-specific transformations which are specific to pathology, for example, stain normalization. Sometimes we need to extract the hematoxylin out of these images, and so on. So I'll just show you some of these transformations which we require. Then these codes should be GPU optimized. So these algorithms should be able to utilize multi-GPU capabilities, good acceleration, and in the case of, for example, A100s, we have MIG capability. So these codes should be able to utilize all those hardware capabilities. The codes should be reproducible, as I've said, and this should be high quality as well. All right, so I will start with the image IO, because as I mentioned, we have huge images, gigapixel images. We need to have the readers for efficient readers for reading these images. These images come in many different formats. Most commonly used format is are based on multi-resolution TIFF images. SVS is so TCG is one of the biggest resource of these images, open repository, and all of these images are in SVS format, which I think now have been converted to DICOM. But all these various formats, SVS and DPI, or from various scanners, they are mostly in multi-resolution tiled TIFF images, but they can be be using various different compression algorithms. So what we need is a reader which can efficiently read these gigapixel images, and these should be supporting the algorithms which are if you're looking at the lower magnification, we are looking at high magnification or intermediate magnification. These readers should be able to read these images efficiently. That's the purpose of that should be the purpose of these readers. So MONAI uses QSIM as a reader. So currently it supports multi-resolution TIFFs, but we are looking at some other image formats as well. For example, we are looking at JPEG 2000 compressed images. We are looking at some proprietary formats as well, but that's a bit tricky because of the licensing, but we are looking into that. And then we are also discussing of supporting the next generation file formats based on ZAR, which is very efficient on cloud-based platforms. So we are looking into that to support those formats as well. And there's a discussion going on in the development group in supporting those various file formats. A strong point about QSIM is that it supports GPU accelerated image processing, which is compatible with scikit-image, which is a very popular library for a Python library for image processing. So there is you can use scikit-image API for applying transformations. You can learn more about QSIM and on the GitHub page, and there's a nice tutorial about QSIM at this YouTube link. which explains how to use QCIM and how it can accelerate the reading of whole slide images. So here, this chart shows the comparison of QCIM acceleration compared to a very popular image reader, OpenSlide, whole slide. This is the OpenSlide supports most of the whole slide image file formats. So this is the go-to library which is used for reading whole slide images. So this shows the amount of acceleration you could get using QCIM. So that's what MONAI is using for reading whole slide images. Then I will talk about about MONAI label. Again, the license is quite open source, quite open here. Now, what MONAI label is doing in this case is it allows you to perform auto segmentation and then allow the users to interactively annotate the images as well. It supports both training and inference of the AI models. We have Nucleic. I will just show you an example how Nucleic works. But what it does, it allows to collect the annotations interactively. So when we are dealing with, for example, millions of nuclei, so we need obviously a lot of data for training as well, which is very difficult to access, especially when we are just time who are already very busy, and it's very expensive to collect all those annotations in terms of time and resources. So MONAI label is has been integrated with popular platforms with existing workflows, which were already in computational pathology. So I would mention QuPath, Digital Slide Archive, and computer vision annotation tool here, which it supports for pathology. For radiology, there are other platforms as well, but I'm not going to discuss about those. So this is a snapshot of integration of MONAI label with QuPath, which is a very popular standalone application for reading whole slide images, for analyzing and annotating whole slide images. This is a snapshot from computer vision annotation tool and with Digital Slide Archive. So I will just show you a demo of how MONAI label can be used with Digital Slide Archive. So you can launch the Digital Slide Archive. So if it is already on your server, you can just log in as admin. And then you can load a Docker container from Histomics, from MONAI label. So you can mention the path to MONAI label Docker, and it will load it in Digital Slide Archive. Right. Okay, so this is how you can run it. So you can open an image in Digital Slide Archive and open that image in Histomics UI. And when you open that in Histomics UI, you can then provide the address to MONAI label. And then you can select a region. You can select a region within the whole slide image. And once you submit the job, it will generate the annotations, and then you can interactively correct those annotations as well. So this is how it has generated those annotations. Okay, so this is Nucleic, which has been integrated in MONAI now. So what Nucleic does is it allows an annotator to select a few clicks. So you can the annotator can click on the inside the nuclei, and it will generate a mask of those nuclei. It will generate a segmentation mask, which is beneficial, especially if you want to do morphological analysis of these nuclei, which is very important, especially when we are dealing with tumor cells. The morphology is very important for downstream analysis. Okay, something which we are considering to integrate further is the annotation of regions. So you can draw a few lines within those regions and apply the algorithms for segmentation of these regions. So this is what we are considering to integrate in MONAI. Okay. There's continuous development going on in if I talk about MONAI pathology. So you can have a look at again project MONAI, and then there is this project MONAI pathology. You can see all the active pull requests, issues, and what's in progress all on this project board. So this is the structure of MONAI pathology at this moment. So you have MONAI pathology at the core. So you have the data, image reader, WSI reader here, which is based on QSIM. I will just show you on the next slide how this is used. We have patch WSI datasets, and you can generate dataset for to run PyTorch models. Then we have some handlers and then metrics, which can calculate metrics once the algorithm has processed the images. But then we have networks which contain the network architectures, and then transforms. These transforms are again linked with QSIM and scikit image library. So you can apply also some color transforms as well, like stain extraction and normalization of stains. So if I just continue on the next page, so the transforms, you can see they are again linked with QSIM, and these are mainly you can use the scikit image API for those transforms. And then for WSI reader, again, it's dependent on QSIM. So if you want to look at the existing algorithms which have been integrated and you want to use the algorithms which have been integrated in MONAI. So one of them, if you look at the project MONAI tutorials, you'll find tutorials in pathology at this link on the GitHub page. So this is something which has been done on the data from Pandas challenge, and this is this algorithm, which uses multiple instance learning, has been integrated in a tutorial has been integrated in the. This is a tutorial which uses MONAI and shows how multiple instance learning can be used in pathology. Then we have tumor detection as well, which has been in tutorials. You can find how you can run tumor detection in pathology. So this image is showing some other type of regions as well, like necrosis, inflammatory stroma as well. But at the moment, in tutorials you'll find tumor detection, but this can be integrated as well based on the existing MONAI core. So with training of the algorithms with different regions. This is this HONET for nuclei segmentation and classification. Eric mentioned that this has been integrated now in MONAI. This is state-of-the-art algorithm for nucleus segmentation and classification. It has won many different awards and many different challenges actually. And it has been, so recently we ran a challenge with the name of Conic, and the winner of that challenge again used a variant of a variation of HONET as well. So this has been integrated with MONAI now, and yeah, I mean, you can use these kind of algorithms. For stain normalization and extraction, there's still work going on. So what stain normalization means is that, so when you get these images, there is variation of stain from when we get these images from different labs. The stains can vary within the lab as well, depending on the batch of the stain you get. So there is some variation in stains when you get these images from various sources. So the deep learning algorithms, the computational pathology algorithms, not just deep learning algorithms, they sometimes are affected by the kind of stain. They are affected by the variation in stains. So what stain normalization does is it can take the stain from a target image and apply it to a source image to normalize the stain. So the efficiency of the algorithms is not affected much. There are many different stain normalization algorithms. Some of these I've shown it on the right side. And then there are some stain extraction algorithms as well. For example, this is a hematoxylin and eosin image. So the purplish stain is the hematoxylin. Purple color, where you see the purple color is more hematoxylin, and then the pinkish color is eosin. Sometimes it is useful to extract the hematoxylin for detection of nuclei. We are also working with integration of, so this is a tool, a toolbox, which we have developed at Warwick, and we have many different computational pathology algorithms. HoverNet is also in this toolbox. So what we are doing is we are working with the development team to integrate models which are part of the toolbox into MONAI, and we are trying to use MONAI framework in the toolbox itself as well. So there's a good integration, there's less duplication of effort, and we can make good use of each other's of these tools. Then we have there's some work going on in open pathology and datasets. So something which you find in built in Torch is, for example, MNIST datasets, CVAT datasets. You can directly download them from using Torch API. So something similar we are looking into in MONAI as well, and that you can use an API and download some of the open datasets. I think Eric mentioned that TCIA data can be downloaded with MONAI now. So that's what we are looking in, and this is again in the discussion section on MONAI, as Michael mentioned. So to conclude, MONAI is for pathology, for pathologists. It provides a starting point for research scientists and pathologists to utilize the benefits of deep learning. It is aimed at acceleration of to accelerate the adoption of deep learning algorithms in computational pathology and provide standardized, efficient pipelines for pre-processing analysis and visualization and collecting interactive annotations for pathology. I've put some links here for tutorials on MONAI. You can find it under tutorials on pathology. All the work which is going on in MONAI pathology, all the open and closed or closed pull requests and issues, you can follow that on the project page. And there's again a working group discussion. We have bi-weekly meetings which are minuted on the wiki page as well, so you can have a look at all this, which is available on Git. That's all from my side. If you have any questions, thank you.
Great, thanks, Shan. We have a few questions. I'm going to go ahead and share my screen because we have some through Zoom and we have some through Discord, so it'll make it a little bit easier to see it if I go ahead and share here. Some of them I don't know. I have some things I can answer on them as well. But we have a question that said, can we use it to draw a bounding box of medical images? I'm assuming it's because when you showed it, you showed sort of the region of interest in DSA and the Histomics UI of sort of the region of interest annotation. So I can kind of answer that one. We have a few different things around sort of bounding boxes depending on how you're annotating. So for sort of CT/MR in, for instance, 3D Slicer, there are annotation methods that you do set region of interest or bounding boxes sort of around an area. You can use either scribbles or grow from seeds, segmentation or annotation methods within something like 3D Slicer. We actually have some videos up on our MONAI YouTube channel that talk about how to go through some of that process in something like MONAI Label. The other thing I was going to mention is that we also had some object detection components that were added recently into MONAI on our 0.9 release. Eric, I don't know if you wanted to talk a little bit about some of this for sort of the bounding box handling at all, or if you know as much about it.
I'm afraid I don't know terribly much about that.
Okay.
I'm about to leave that to others.
Okay. So just to kind of answer that, there are some in there. I think there should be, usually there's a tutorial for each of these. So there should be a tutorial around here. And we do have modules for some general bounding box sort of handling in medical images. But it really depends on what you're looking to do exactly. So next question, do all the applications or viewers, QuPath, DSA, and CVAT support active learning? Shan, do you know if any of them right now support active learning? I don't know if we have any of the active learning for pathology right now. Do you know?
I'm not sure about that, but I can check with Sashi, who's developing this.
Okay. I know just generally for MONAI Label, and I'll talk a little bit about this later on as well during our roadmap section, is active learning is something that we're kind of doing research into right now. A lot of different places are trying to do active learning because we're seeing a lot of success with active learning. So even things from video streaming for things like endoscopy, that's a little bit different of how you do active learning on that sort of data set versus something like pathology versus something like CT/MR. So hopefully we can get the answer for that one. But just generally, there's a lot of, in the next few releases for different frameworks that we have, active learning is a big thing that we're focusing on. I'm actually going to jump down to number 8 here really quickly. So what is the average prediction of cancer mitosis with less number of training samples? Shan, I don't know if there's quite an answer you can probably give to that. I don't know if you wanted to give any just general information about sort of training samples and some of the predictions you've seen in working with
I think it depends on, I think this is, if this question can be very specific, that would be better. But I mean, it depends on on the model as well, and the kind of data we are training. There are still, in this question, there are still a few variables which needs to be answered. Yeah. So, I mean, my answer cannot be very clear on that.
Yeah. I'll jump back up to number 7 really quickly. So for DICOM images, so you can use DICOM images generally. We have DICOM readers in, for instance, in MONAI Deploy. So if you wanted to use it just for the deployment piece, there are DICOM operators in there, and you can convert things over to Nifty as well. It really depends on which piece and whether we have operators for there, but we have support for a lot of different IO readers or custom IO readers, depending on what you're exactly looking to do. A few other questions came in. So it seems like MONAI could provide auto annotation for pathology images. Should I provide some prior information about what I'm looking for? Let's see. Is there any official pre-processing pipeline for pathology images in MONAI? That was sort of maybe that's kind of a follow-up. I don't know. I don't know exactly. Maybe if you can expand a little bit more in Discord about what you're exactly looking for. For pre-processing, Shan, you talked a little bit about some of the transforms that were there. Is there any sort of specific pre-processing pipeline that we suggest, or that we have some examples of or tutorials?
I think I'll have to look in the tutorials. But again, pre-processing also depends on what kind of model you're using. Because some models use pre-processing and that their preprocessing is necessary to run those models. But for some of them, they just can take any ordinary RGB image and just run those models.
Yeah.
There's one question on in the chat about NuClick. NuClick uses pre-trained segmentation model, and that's most probably trained on pan-nuke dataset.
Yeah, so that was this one here, is NuClick functionality based on pre-trained. Yeah, cool. Are there any plans to support IC or image J, which are used for cell visualization?
I'm not sure about that, but QuPath is, I mean, for standalone applications, QuPath is what we are supporting, what MONAI is supporting. And for web-based, we have computation CVAT and also the digital slide archive.
Okay. So for digital pathology, it's seen a lot of synthetic data. How is MONAI Pathology module looking forward to contributing in synthetic data generation in digital pathology? I know that, I think, Eric, you actually had, I don't know if you had looked at it in pathology, but I know that you've done some, I think, about synthetic data generation and some discussions around there. Did you want to just mention generally sort of MONAI and synthetic data discussions?
Well, synthetic data, a lot of what we've been doing has been augmentation. So that's kind of synthetic data in a sense. So we will modify images a lot during training. We have some synthetic data just for doing testing on, really. That's mainly what we've been doing. But for doing actual generation of data that's supposed to represent actual CT or MR, for example, that's something that's sort of been other areas of research we haven't really gotten into terribly much.
Okay.
Yeah, so at this stage, there is no synthetic data in pathology in MONAI, but I think this is a good idea. We can discuss in the development meeting group. Yeah.
These are the sort of bundles we do want to get into, networks for generating data, GANs or VAEs or something of that sort. And I think that's going to be an area of future research that we would like to see contributions from.
Yeah, definitely. So someone asked, how does NuClick identify the segmentations of different classes? In the video for NuClick, saw that by clicking on the cells, identified the cells and labeled in different colors.
So
what is it sort of doing to do that?
These labels were mainly different cells. It was just identifying different cells, but there is a way, so you can before you click, you can select a different type and then click. That will be saved as a different label, as I showed for the regions. So you could see that the annotations were different colors, and then you could have different colors for regions. But the demo was mainly to identify different cells there, that they were different cells. Yeah.
Yeah. And then generally, I think this kind of goes back to the active learning, if you want to add anything else, but generally for 2D active learning, I don't know, you said you'd have to reach out to Sachi, I think, to confirm what sort of active learning is being implemented for pathology. So I don't know if you have any other answer than what we kind of talked about before. Okay. So I think, so I don't know for sure what we can get back to you about what other pieces of active learning that we have in there. If you open up a discussion on the MONAI label GitHub, we can answer it on there as well. So it's something where you can definitely go over there and ask it. If not, I can try to reach back out. Message me on Discord or message me on Slack, and I'll see if I can find you an answer for that. So for people who have been asking those things, feel free to reach out and I'll see if I can get you an answer sometime after the event.
And for anyone who would like to contribute to, and someone has mentioned that in Discord, if you can open issues or open a discussion on the main MONAI repo or whatever repo is appropriate, we definitely pay attention to these things, and anyone who'd like to contribute, we can start the conversation from there.
Definitely. One last one that I did see is MONAI completely dependent on GPU? The answer to that is no, it doesn't need a GPU, but it will be a lot slower to do any of the training. It really depends on what you're looking to do. For some of the inference pieces, if you're just doing a single inference, a CPU would be okay in something like deploy. But yeah, training, it already takes sometimes, depending on what you're doing, hours or days to be able to train some of the networks on a fairly decent GPU. So if you switch it over to the CPU, you may see a 5 to 10x or more increase in time to actually do some of that training. But it is not specifically dependent on GPU. I think we got through all the questions. If I missed one on accident, let me know or repost it in one of the Q&As. But again, thanks, Shan, for going over all of that.
Thanks, Michael. Thank you.
All right, so next up for our agenda, we have Seyed-Ahmad Ahmadi from NVIDIA. He's going to be talking about large-scale pre-trained models, specifically SWIN UNETR. So Ahmad, go ahead and give yourself an introduction and then take it away.
Hi, everyone. My name is Ahmad Ahmadi. I'm a senior solution architect with NVIDIA. I work, my main domain is deep learning in healthcare. And I had a lot of academic work up until last May when I joined NVIDIA. Basically my main working area is with MONAI, which I'm very happy about because it's a very lively and exciting framework. Today I'm going to present some very recent work on self-supervised pre-training using vision transformers. Let me share my screen for this. Can you see my screen?
Yeah, we can see it.
Okay, great. So I'm the presenter today for this work, but I'm not the originator of this work. So I'm have the pleasure to present this on behalf of my colleagues at NVIDIA's DLMET team. This is sort of the research team for medical imaging at NVIDIA. The main author, Yu Cheng Tang, first author, and under the supervision of Ali Hattamizadeh. This is the work on self-supervised pre-training of SWIN transformers, and what this means exactly we're going to get to in this talk for 3D medical image analysis. This was a paper at CVPR, and kindly Yu Cheng gave me his slide deck from the CVPR presentation. I hope I can do his this work justice today. So the goal is medical image segmentation in this work. A lot of the scenarios in MONAI, a lot of the tutorials, a lot of the entry examples in MONAI are centered around medical imaging. And this particular example is about medical imaging using vision transformers. Medical imaging, there are two data sets that are considered in this work, whereas one is beyond the cranial vault, BTCV. This is basically, the task is multi-organ segmentation in abdominal CT. And the other data set are actually 10 data sets. It's the medical segmentation decathlon. Many in the audience will be familiar with this. It's basically a suite of 10 different pretty challenging segmentation tasks on various anatomies and modalities, brain tumor and MRI, heart and MRI. A lot of tasks for abdominal CT and lung CT segmentation, hippocampus segmentation in the MRI, and prostate segmentation in MRI. So in essence, you can say it's sort of actually 11 data sets that are where this work was evaluated on. And sort of they also gave a good range, together they give a good range of the different, of the variety of tasks in medical image segmentation. And you might say that this is already a solved problem. There are really strong methods out there. I think everyone on the call is familiar with UNET, which was proposed in 2015. And of course, this is also available in MONAI, just because in the very beginning, I heard that there are, I think it was a good 50% who have not used MONAI yet. If you're not familiar with the tutorials repository, usually I recommend to get started with MONAI using this tutorials repository. There are various scenarios in this repository. If you go to one of the scenarios, 3D segmentation, right off the bat, the two first tutorials are basically on BRATS, brain tumor segmentation, spleen segmentation in 3D. Of course, with a 3D UNET, there are also other architectures, UNET-like architectures in MONAI implemented and readily available. Dynamic UNET, particularly, which is sort of a highly customizable and parameterizable version of UNET. So this is, of course, there are tutorials here for spleen segmentation, how to get pretty good near state-of-the-art segmentation results on these data sets. Of course, this is there, but there is, of course, the motivation that there has been a recent line of successes of vision transformers in computer vision. And we would like to see whether this can be applied to and sort of transferred to the medical imaging domain. But then there are, of course, challenges. So first of all, 3D, most of these vision transformers in computer vision are in 2D, and they are sometimes pre-trained on large data sets in self-supervised manner or supervised manner for classification at least. But there's a large domain gap between natural images and medical imaging modalities. Also, if you have 2D models, you have this problem that your inference results lack the cross-plane contextual information. So you get discontinuities in the z-axis. On the other hand, we have the problem medical imaging that generating expert annotations to do fully supervised training is pretty expensive. It's really non-trivial. You need experts for this, and which makes it so expensive, and is very time consuming because 3D annotation is usually more work than doing it in 2D. Another challenge in medical imaging is that we have these very large volumes, but potentially very small structures that are somewhere in the 3D volume. And we also have partially long-range 3D spatial context, which should be somehow encoded by these vision encoders. So vision encoders often in computer vision are for classification, where you can encode the 3D spatial context over a large field of views, but you don't need to recover fine structures for segmentation. So there are certain challenges. And there was previous work also in MONAI, also from the DLMed team, a contribution to MONAI from the DLMed team and NVIDIA, the so-called UNET transformer, the UNETR architecture. It's been in the repository of MONAI, I think, since 2020 or late 20, I think. So already available for quite a while. Basically, the basic idea is that a UNET transformer architecture consists of a transformer encoder. It directly utilizes 3D patches, and those 3D patches get encoded together with some spatial encoding, and that gets forwarded to the encoder result basically gets forwarded to a CNN-based decoder via skip connections, and the CNN-based decoder, very similar to a UNET, to the decoder branch of a UNET, then creates the segmentation output maps. If we take a look at this in a little bit more detail, so if you have a 3D volume, just like in regular vision transformers, that 3D volume gets divided into a sequence of uniform and non-overlapping patches. These patches get projected linearly into a linear embedding space with this sort of linear projection layer. And then to the sequence, positional embeddings are added in order to encode where this patch came from in the volume. And this is then used as input to a transformer model. The transformer model basically uses its multi-head attention mechanism to establish spatial context between these patches from different regions of the image or the volume. And these encoded representations from the different resolution layers. So the resolution idea also is very similar to the unit encoder idea. So the encoded representations from these different layers, they get extracted and skip connections forwarded and deconvolved into higher resolution layers, and then basically the CNN decoder branch takes care to reconstruct a segmentation output map. Now, the problem is that with vision transformers, because you have to extract these non-overlapping patches, and you want to establish pairwise multi-head attention between all patches, you can only use so and so many, or your patch size has to be fairly large in order to be able to fit this into VRAM because of the quadratic complexity of this multi-head attention step. And this is inherently a problem because we want to actually have also very fine-grained representations at the finer layers. And this is, you can also see it here, it's basically always divided by 16. So it's you take your input volume and always divide it by, or you create 16 by 16 by 16 maps, and not much higher than this. So the SWIN UNETR architecture is very similar to the UNETR architecture, but it follows this multi-resolution step a little better. So in the fine resolution layer, you actually have quite a lot of patches that are that encode very fine-grained structure and local structural spatial context, whereas in the lower parts of that pyramid, you get increasingly larger blocks or subblocks of the volume for to learn the multi-head attention. But similar to the UNETR architecture, the encoded representations get forwarded to a CNN decoder towards the segmentation output map and the loss for segmentation. There is a very crucial difference to the UNETR architecture though, and this is this shifted window self-attention mechanism, which is why it's called SWIN UNETR. In these transformer blocks, we don't have just multi-head attention, but we have two steps of multi-head attention. One on the window multi-head self-attention, and then on the shifted window one. Why? We can see this in the next slide. So the idea is that instead of having fairly large blocks in order to be able to fit the quadratic complexity into your GPU memory, instead of doing this, we can keep very fine-grained blocks, but then we have to limit the spatial context somehow. And this is this idea of windows. So instead of having multi-head self-attention from every block in this volume to every other block in this volume, which can get quite large, we subdivide the volume into windows, or we group patches into windows. In this case, instead of having 3D tokens of 8 by 8 by 8, with we take basically 2 windows in each dimension with 4 by 4 by 4, which means that we have 8 windows that represent this volume, and we only compute multi-headed self-attention from each patch in this window to each other patch in this window. So in doing this, we basically reduce the quadratic complexity to a linear complexity, because we don't have from every to every other, but basically you can say the receptive field of this window, the patches are limited. The only problem is that if we compute multi-headed self-attention within a window only, then we lack the spatial context of, for example, this patch to the patch over here. So there's sort of no cross-communication between these windows. And for this, there is every SWIN transformer block has 2 multi-headed self-attention parts. So basically these blocks always come in pairs of 2. You can have 2 or 4 or 6, but you cannot have 1 only, because the 1 basically helps us to maintain a linear complexity, but we want to have the cross or the long-range spatial context. And for doing this, the next layer of multi-headed self-attention is again on windows, but these windows get shifted by 2 patches. And you can see this here. So basically now the center patches can compute their multi-headed self-attention to each other, bearing in mind that they already have established their multi-headed self-attention in the previous layer. So we sort of get this similar to how we do convolution with strides. We increase the receptive field of the multi-headed self-attention by shifting the window by 2 blocks. And this is the second layer of the multi-headed self-attention with the shifted window in order to establish the longer range spatial context. This is the one trick in this work, introducing the concept of SWIN transformers, which was proposed by Microsoft researchers in the computer vision domain, but now fully in 3D for 3D vision transformers, and by extending the previously proposed UNETR architecture. Now, this work has another key contribution, which is to pre-train a large SWIN transformer encoder using self-supervision. This was done in this work on 5,050 CT volumes from different data sets: Luna 16, TCAIA, COVID-19 and colon, LIDC, HNSCC. And basically, these are data sets of CT for different areas of the hip upwards. So we have abdomen and pelvis, there are thoracic CTs, head and neck CTs, and whole body CTs across these volumes. And there is, we will come to this later, there are, this SWIN transformer encoder was pre-trained on 96 by 96 by 96 crops from these volumes, random crops from these volumes, and in order to learn a strong transformer encoder that can then be fine-tuned towards downstream tasks by basically fine-tuning the CNN decoder. And the self-supervision mechanism, I mean, there are a lot of recipes out there for self-supervision. In this work, there were 3 pretext tasks that the SWIN transformer encoder had to learn towards self-supervision. These pretext tasks was that from the input CT, we basically take a subvolume. From the subvolume, there are different augmentations that corrupt these 2 blocks. So there are, for this, we take this block and we basically augment it in 2 different ways, so that we get 2 different corruptions. On top of that, we also rotate one of the 2 blocks. And the rotation is along the z-axis in 4 variations, either 90 degree, 180 degree, 270 degree, or of course 0 degree. And these are basically 4 classes. And this is one of the downstream tasks that the self-supervision pretext task has to, or this network has to learn. So this is one component of the losses. It's basically cross entropy. It's a classification by how many degree this was rotated: 0, 90, 180, or 270 degrees. The other task, pretext task, is in painting. So we get corruptions by basically blocking out certain subvolumes and subblocks of this volume, and these need to be reconstructed. So it's basically an in-painting task. The loss here is MSE. And there's also a contrastive loss. So the encoder representation basically gets passed through another linear layer. In this work, it was 512 dimensions. So we basically get a 512 dimensional representation of this block and of this block. And then there is contrastive learning, which has to learn that if these, so there are random pairings of these blocks, sometimes from the same volume, sometimes from, this is from one volume and this one is from the other. And in this contrastive learning, the network has to detect whether, or has to align, maximally align the embedding vectors of patches from the same volume and maximally make dissimilar the embedding representations of patches from different volumes. And this is done via cosine distance. So these are 3 different losses. They are weighted equally with towards a compound loss. So it's a multitask learning problem during pre-training. And that basically creates a fairly strong encoder, and how strong we will see in the next few slides. So as I said, this method was applied to the BTCV abdominal multi-region abdominal CT segmentation task and the medical segmentation decathlon benchmark. And in the BTCV challenge, across the different anatomies that are segmented, SWIN UNETR, after pre-training with self-supervision and then fine-tuning on 24 labeled volumes in the BTCV challenge, comes out on top on most of these anatomies, and on average also it comes out on top. And in the medical segmentation decathlon, It also comes on top compared to other pretty strong architectures, including DINS, which is differential neurotopological search, so an AutoML technique that is also implemented in MONAI and readily available, and another very strong model, known UNET, which is probably fairly known in this audience as well, published in Nature, and a really strong method from the German Cancer Research Center. When you look at the challenge leaderboards, you can actually see in the mean position on the tasks from the medical segmentation decathlon across the different submission ordered by average rank on these different tasks. There's a huge drop that was achieved by known UNET. And UNET, basically UNET 3D vision transformers, the UNETR architecture does have a little further gain, but with self-supervised pre-training, there's really a significant gain using the SWIN UNETR architecture. Likewise, in the BTCV abdominal challenge with average dice on the different regions, again, one big jump among the submissions ordered by average dice, one big jump with known UNET, some small gain with vision transformers, but then a big jump with a pre-training on these 5,050 volumes and the 3 pretext tasks that I described. So really significant advantages. There are also qualitative comparisons in the paper. If you look at the ground truth, for example, here for pancreas segmentation, the SWIN UNETR gets certain regions that other methods are kind of struggling with. Even though UNET, which I think is a very strong architecture and approach, likewise on very thin and elongated structures like this portal vein segmentation in the liver, again, some of the methods struggling a little bit, and SWIN UNETR can keep this, can recover this basically due to this long range spatial context that the encoder learned. And on adrenal gland, a similar situation where the SWIN UNETR follows the ground truth more than the others. There are a lot of comparisons on the MS medical segmentation decathlon task. I won't go into them now in detail, also because it's important to zoom in. Feel free to look into the paper. In the paper, it's compared to DINS, this AutoML technique that is similar to known UNET, basically automatically parameterizing a 3D UNET architecture towards the downstream task. And there are comparisons, qualitative and quantitative, to that. So there are there's also a very nice ablation study in the paper of SWIN UNETR. First of all, it's analyzed that on how much, if you take the percentage of labeled data in the BTCV dataset with only 10% of labeled data up to 100%, which is basically the 24 volumes in the data. In the training data, because BTCV is actually not a very large data set. If you compare training from scratch to pre-trained segmentation using the pre-trained encoder, SWIN UNETR encoder, you get much better performance, especially with fewer volumes. And in order to reach something like the best target dice of the training from scratch on the full data, you need around 40% less data if you have a pre-trained encoder branch. There are also in the MSD CT tasks, there are significant improvements on all the 6 different CT-based tasks. It's important to note that for the MRI-based tasks in the segmentation decathlon, the SWIN UNETR was trained from scratch, not with pre-training, because there's too much, the domain shift is too large. But on the CT tasks, the pre-trained encoder was possible to use. There's also an ablation study of the pretext tasks, which shows that among these, if you only use one of the 3 pretext tasks, in painting is best. But across the board, if you basically combine these 3 different pretext tasks, then you reach the best performance. And of course, this is also a nice training ground, or like a nice starting ground for self-supervised learning research, which you can basically use this repository for. Scaling of pre-training data. So this is an analysis, how much pre-training is necessary. I told you that that it was pre-trained on 5,050 unlabeled data samples, but this was also analyzed for 3,000 only and for 100 only, and compared to training from scratch. And you can see that training from scratch on BTCV, basically, because it's only 24 labeled training volumes, which is a very realistic case in medical imaging. Having 24 multi-region abdominal CTs fully labeled by radiologists is actually a fairly luxurious situation, but it's really not that much in order to train a model from scratch. Whereas if you do the pre-training, you get better improvements. And the bottom line of this analysis is basically that it really benefits from an increased size of unlabeled data, which is also a big potential, because typically, especially clinical research centers have years and years of archived data, radiological scans in their PAC systems. And this analysis basically shows that it's not only possible to use this data in an unlabeled manner, but it's actually beneficial to use as much as you can to pre-train your encoder branches. Now, this is all the method and the background. How can you get started with this? So you can go to Project MONAI, the GitHub project page of MONAI. There is a repository called Research Contributions. Inside Research Contributions, there is a folder called SWIN UNETR, and there are 3 folders. One is for pre-training, one is for BTCV, and one is for BRATS. BRATS is basically training from scratch, just a SWIN UNETR, because the pre-trained model was on CT. The pre-training is explained there and fully coded out, actually fairly conveniently with a script where you have quite some customization options. You can, for example, specify the region of interest. It was pre-trained in the paper on cubes of 96 cubic. But you can also increase this or decrease this depending on your hardware that you have available, or depending on the resolution that you want to cover in your target data. This script is already enabled for multi-GPU pre-training, but you can also do it on single GPU if you have limited hardware. And if you don't have very large GPUs, then you can even activate gradient checkpointing, which allows you to get away with smaller VRAMs. But the paper was trained on 8 32 gigabyte V100 CPUs, basically a DGX node. And the code is prepared in a way that you can leverage such an architecture if you happen to have it at your research institute. A few notes. So when you set up your own experiment, you again use a research collaboration repository. I think you have to use a particular version of MONAI because that was the implementation of the SWIN UNETR at the time of publication. So I think it was version 0.81, but feel free to check that. And you can always use the containers on Docker Hub to basically reproduce that setup that was also used in the paper. So use the Docker Hub container with that tag. Prepare your data set in the subfolder of pre-train. In utilities, there's a file called data util. So you just have to basically exchange the data and create your own data list if you want to use your own data. Think about your transforms and model parameters. Again, use 96 cubic if you want to later use the pre-trained models, or if you want to be in alignment with the paper. A few notes about GPU requirements. I tried it briefly only. So if you have 96 cubic crops and you want to pre-train, at a batch size of 1, you need 8.4 gigabytes of VRAM, so which is a fairly realistic size for many GPUs in academic centers. For a batch size of 2, you need 15.5 gigabytes, and for a batch size of 4, which was used in the paper, you can nicely utilize V100 GPUs at 31.2 gigabytes of VRAM. The training duration roughly on 4 GPUs for 100,000 iterations is 32 hours. So, and of course, multiplied if you use less GPUs, but it's basically a few days of pre-training in order to create your own custom SWIN transformer encoder for your own data set, let it be CT or anything else. Once you have pre-trained this, or if you want to use the pre-trained models that were used in the paper, these are also available. So for the BTCV data set at least, or for the CT pre-trained encoder, again, go to the research contributions repository, to the BTCV subfolder. You can download the self-supervised pre-trained weights from SWIN UNETR backbone in the CVPR paper. There's the link for this. You can even download the fine-tuned ones on the BTCV challenge. So you can get started with a SWIN UNETR that is capable of doing multi-region abdominal CT segmentation. There are 3 models available, one with a feature size of 48. So basically the embedding size for each patch in the SWIN UNETR encoder layers. There is also a smaller SWIN UNETR with an embedding size of 24 and a tiny one with 12. The according dice overlaps are here. So especially the small one, if you have smaller GPUs, is still fairly good, has still a fairly high fidelity in terms of segmentation. Unfortunately, there's no self-supervised pre-trained model available, but for the base one that they also used in the paper, the pre-trained, this dice overlap was achieved with the self-supervised pre-trained model. And again, these are all available for download. And then there are also recipes on that page. It's in the README file, concrete commands similar to here. So the concrete calls to the training script or fine-tuning script, either train from self-supervised weights on BTCV or fine-tune these BTCV models from the paper on your own data set, or training from scratch in case you don't want any pre-trained model. So these are all there, again, for single and multi-GPU, so fairly convenient to get started for you. And this is already the end. Just as a conclusion, SWIN UNETR self-supervised pre-training. SWIN UNETR itself is a transformer that is a strong encoder which benefits from large-scale 3D medical image pre-training. The self-supervised pretext tasks, the 3 pretext tasks, can benefit or leverage this unlabeled data. And in this work, it's also shown that this unlabeled medical images carry valuable data. Self-supervision allows you to train on them without any additional annotation cost. There are multitask learners for self-supervision, and this is already implemented. And again, I think it's good to look at this as a great starting point for your own self-supervised learning research. You can easily extend this architecture, introduce new pretext tasks or modify them. One thing that the work also shows is that it's beneficial to scale up your data sets. And if you do so, then you can achieve state-of-the-art performance. This is what the authors showed on BTCV and MSD challenge. Of course, it reduces the burden of manual annotation. And as I said, I would say, consider this a starting point. The scripts basically work out of the box on the pre-trained models, but of course you probably want to modify this on your data, on your own data, in your own modalities. So consider this a starting point. And in alignment with the first talk of this morning, I see this also as a great opportunity. I'm pretty excited to see how the community will use these resources and hopefully create large self-supervised pre-trained encoders that can then be shared in the model zoo for community collaboration. So I hope this was helpful today to get you started. And please reach out if you have any questions, either today in the chats and Discord, or also on GitHub in the issues or in the discussions of the research repository. Thanks a lot.
Great, thanks, Ahmad. We actually have quite a few questions. I don't know whether we'll be able to make it through all of them, but I know that you are in Discord, so I'm going to go through a few of them just so we can kind of stick on time. And let me go ahead and share really quickly.
Yeah.
And,
should I unshare?
I think I can take over from it. So I think I took over the share there. So I'll just start with the top. Some of them you already answered later through your talk. So some of them will be easy to skip over, but I'll just start from 16. When I use SWIN UNETR to train my data without using smaller resampling size, is there any other way to train with smaller GPU memory?
Yeah, and I mentioned this briefly. There is a technique called gradient checkpointing. What it exactly means is a little bit too detailed now to go into, but it basically is a technique that allows you to get away with less GPU memory. And this is actually a flag in the call to the main function. So you don't even need to implement it; it's already there. Just activate that flag, and then you can use smaller GPU memory.
Great.
Sorry, I'm going to butt in here a bit. Seyed, sorry, have we tested the network with automatic mixed precision? because that's another technique people could use to reduce memory. However, it doesn't always work 100%. It's a bit, it depends. So
it's true. Automatic mixed precision is also in there, but there is a specific hint in the at this parameter in the when you look at the arguments for the command line call, that you should not use AMP for training. I'm not really sure why. Would be interesting to ask the authors, but it's basically recommended to not switch this on for training.
Okay, we're going to have to circle back to that because we've encountered this with other networks, and that's something we should figure out, because I think it'd be very helpful.
Mm-hmm. Okay. Next question was whether this supports in addressing the partial volume effects, especially if there is a nonlinear mixtures present in subvoxel levels.
That's a very good question. I think the answer is similar to UNETs. Basically, it's a UNET decoder, a CNN-based decoder. The partial volume effects, it depends on how good your resolution is of the output map. Of course, the output segmentation cannot be better than the voxel resolution that you're putting in. I would say the way to maybe deal with subvoxel level effects or partial volume effects is to something that you can do, for example, in Slicer, and that I like to do is to create a surface model out of the segmentation and smooth this a little bit. That's one way to deal with this, to create basically contours that that cross voxels in a diagonal manner, for example, in order to partially deal with this. But in essence, because you're creating these embeddings for each patch in the volume, you somehow encode the patch information and the information of all voxels and how they're spatially aligned in this patch. You're basically encoding this into an embedding. So I cannot answer this directly, but I would say intuitively that it kind of takes this into account by learning how to embed many patches during training, including partial volume effects in there. So these are two very vague answers. I think it somehow addresses that, but I don't have a concrete answer whether it solved this.
Okay. Hopefully that was a good enough answer. If you have any other questions, definitely feel free to leave them in chat if you want to mention anything else. We'll do 3 more questions here really quickly. So do you think other methods besides SWIN UNETR can benefit from the proposed unsupervised pre-training technique? Yes, definitely.
And there are also other self-supervised pre-training techniques. If you go to the tutorials repository, there is a folder for self-supervised pre-training, and there are other recipes in there as well. So if you could do this really quickly, just go to the folder self-supervised pre-training. There it is, and there's a self-supervised script and fine-tuning, which is not SWIN UNETR, and it's trained in a different way. So again, these are several examples in MONAI how to do self-supervised pre-training, and use these as a starting point to do your own research and try to implement the recent state of the art, ball of twins or whatever. And if you do, please share it with the community.
Great. Let's see. Number 19, was this network used for in-painting as well? Can you please expand more about the performance obtained on this task?
Yes, it's one of the pretext tasks. So one of the 3 tasks. And it does this, but I have to say that the reconstructions that you get do not look very good. Yeah, and I think that's also not that important for the pretext task of in-painting, especially because it is one of the shared 3 tasks, and they are weighted equally. So the contrastive learning, the rotation, classification, and the in-painting are weighted equally. So the reconstructions that you get are not looking that great, but it's enough to train a pretty strong encoder branch.
Great. We'll do 20 as this last one. I think we got through all the ones that were not in Discord, so I think the remaining questions are in Discord. So let's do number 20. It seems like you try the large-scale pre-training by contrastive learning, but its performance of downstream tasks mainly depends on the augmentation methods in pre-training. Do you think trying the newly supervised method in MAE by K-mean HE is a good idea?
Yeah, definitely. And I think I've even seen a MONAI based work on this. I'm not sure whether the code was publicly available, but it's a different way to do it, and I think it's a very good idea. And again, please do it. And if you do, share your code, because I think that other work that I saw did not have a public repository. But all these recipes, I think it's great. It's MONAI. The idea of MONAI is also to have a platform where we can share these things, and so that we can learn from it together. It's a very good idea, definitely.
Definitely. I'll go ahead and answer. It's 21. You did share that there were the weights on there. You showed where the location of them were. So you can go get those, and they have sort of the dice score and the different links to each of the different models that were on there for the weights.
Absolutely. So if you do CT on anything abdominal or upper body, including head, I think you have pretty nice pre-trained weights. Great.
And so I think we'll leave these other three since we're a little behind to Discord. But Ahmad, thank you so much. There was a lot of good discussion around this, so I'm glad you were able to present today. And hopefully you can get to some of the questions in Discord.
Yeah, great. Thank you.
Cool. Next up, we have Thomas Roberts from KCL. He's going to be talking a little bit more about MONAI Deploy in the clinic, specifically AIDE and their AIDE platform and how they're deploying those out to trusts with the NHS. So Thomas, I think you should be able to share your screen. Feel free to introduce yourself and give yourself an introduction, and then take it away.
Sure, thanks, Michael. Let me just try sharing my screen. Windows here. Can you see that? Okay.
I can't see anything yet. Try maybe one more time.
Wait. Oh, yeah, sorry, I realized what's happened.
There we go. You can see it now.
Okay, thanks, Michael, for the invite and for also arranging all of this. Much appreciated to you and your colleagues. So I'm Tom Roberts. I'm an AI engineer in London in the UK, and I work for one of the hospital trusts here, namely Guy's and St Thomas NHS Foundation Trust. And I'm also presenting on part of the behalf of the London AI Centre as well, which is a consortium in the UK to do with AI and deployment in hospitals as well. So I'm going to be talking about AIDE, which is the AI deployment engine, and how we're using MONAI Deploy in the clinic. So this is an implementation of MONAI Deploy. And just to give a bit more of a background about me, I used to be an MRI physicist as well, so I'll be referring a bit to that in my talk. And so I've got a lot of experience of working in the clinic alongside doctors, also computer scientists, technical people as well, and trying to link up those two different domains, basically. So this is going to be quite high level in that regard, talking about a lot of these different experiences. So I'm sure we're all familiar with how much of a sort of boom in AI there has been, and how much investment there's been, really. So these are various different news articles talking about all of the investment that there has been made in different AI technologies, all parts of the AI lifecycle, basically. So right from development through to deployment in hospitals, and then also looking at post-market sort of monitoring of AI and things like this. But what we find is that there's a real gap between all of this innovation and also the uptake in hospitals. And so on the left here of this diagram, this green schematic is representing everything outside of the hospitals. So this is universities, this is technology companies working in AI, and there's a really large skill set here, and there's a lot of technology available in all of these places, and a lot of investment into all of this as well. But then there's a real gap, a chasm between getting these technologies into the hospitals, and part of this is because there's just not much investment in the hospital side. So there's not much. There has traditionally never been much motivation to invest in people to understand how these AI technologies work. There's not so much work on the policy side of things, and this is a really important area for hospitals. So What do you do if an AI goes wrong, for instance, in a hospital? Or what biases are there? How do the hospitals prepare processes to deal with these things? And this area is improving. There's increasing amounts of sort of guidance about AI and different papers to do with AI, government papers. But this is still lacking to some regard. And then what I'm going to talk about today is that there's no real common platforms for delivering AI solutions into medicine. And what all of this amounts to is that ultimately you have all of this investment and all of this technology, but on the hospital side, you don't really have any value for the patients. You know, we're doing all this amazing research effectively, but if you go to the hospital, you're probably not going to find many cases at the moment, at least, where AI is being used to deliver your healthcare. So we're not actually delivering them value. And as a sort of exemplar of that, I mentioned at the top of this talk that I was an MRI physicist. In my old department, when I worked at King's College London, we spent 15 plus years working on, and we still are, motion corrected 3D fetal MRI. So on the right here, we've got a sagittal projection through a mother's womb, and you can see a little baby moving around inside the womb. And what this means is that your images can very easily be corrupted, your MRI images. And so at King's, we were developing techniques where we would acquire multiple stacks of different MRI images through the baby's brain. And then, let's skip forward. We could perform AI-driven 3D motion-corrected reconstructions of the baby's brain. And between 2021 and 2022, at our hospital, Guy's and St. Thomas, we performed more than 750 of these AI-driven 3D fetal reconstruction. So the technology is there, and the investment has been there for multiple years. But the problem is, we are only using this technology at Guy's and St. Thomas Hospital. I'm very passionate about trying to get this into other hospitals. So this is this big problem of using this more widely and delivering value to patients. And that's not just true of university environments as well. You know, there's many challenges for getting industry developed AI technologies into hospitals as well. So from a platform perspective, I just wanted to sort of discuss some of the different challenges for medical AI. So on the deployment front, as AI becomes more and more complicated and more sophisticated, and there are more and more AI models, that becomes an exponentially increasing problem. And so that's something that hospitals have to manage. You know, they have to start thinking about, how do I manage all of these different types of AI? And then there's also an interoperability challenge as well. So in healthcare, you have many different systems, many different databases. They might be developed by different companies. They're designed to communicate in some ways, but they might not always talk properly together. They're very heterogeneous, and it's very difficult to link up all of these different technologies. So that's a real challenge as well. And once you throw AI into the mix, that becomes even more complicated. And then there's an ongoing problem of evidence generation. So with these different AI technologies, we need to be able to monitor their performance as we use them and as we go through the future. So we need to be able to provide evidence to the hospitals that yes, they are working and yes, they're performing adequately, or find out when they're going wrong and perhaps why they're going wrong and investigate that. And so from a singular sort of hospital point of view, when we come to deploy AI, there are a couple of issues as well. So just to sort of talk even more about these, you've got point-to-point integration issues. So you can imagine that if you deploy a single AI, you deploy multiple AIs, you have to manage each one of those AIs, and that burden will fall on one of the admins that has been appointed to manage that AI in the hospital. And again, we want to use many different types of AI, so how do we manage all of those things? You can also imagine that if you have an AI product that is installed in a hospital, it might come with some kind of potential hardware that the company wants you to use with that AI because it's optimized for that hardware. The hardware meets the standard of the AI. But then you could also have a scenario where another company wants to put in a piece of hardware, and another company wants to use the cloud, or another company wants to use a separate piece of hardware. So you will have all these resources on the hospital site but they might only be used for that one piece of medical AI technology. So there might be periods where they're dormant, for instance. So those resources are not being used. And again, that's kind of an underinvestment scenario. And then with all of these different types of AI, I've already alluded to this, really, there's very complex governance around all of this. How do you manage all of these different AIs? And how do you actually deploy them at hospitals as well? There's a very lengthy process currently, really, to go through getting AI deployed into different hospitals, because each individual hospital has its own departments to decide how they want to deploy these AIs into their own hospital. It's not unified across the hospital system, at least in the UK. And so that's a very complicated process to get widespread adoption of different AI technologies. So the focus of my talk today is on AIDE, which is the AI deployment engine, which is an enterprise platform that we're creating that will hopefully try to address some of these problems. And so on the right here, I've got a schematic of how it works. So this little image in the middle is effectively AI, which is I'll show you the hardware for that in a second, but the hardware will be hooked up to the imaging modalities at the hospital and also the patient information systems such as PACS. And data will be able to send from those systems to AIDE. And then within AIDE, there will be a whole host of different AI applications which will receive that data and then automatically decide what to do with it, perform an inference or some kind of other AI task, and then pass that data back to the imaging modality or the PACS system. And so to start with, AIDE will be working with DICOMs, but it will also be working with non-imaging formats. There's going to be an app store as well, an AIDE app store, and I'll show you that in the demo later in this talk, a bit like, say, the app store that you might have on your phone, so that different hospitals can choose which applications they want to deploy in their trusts. We're going to bake in certain information around managing these apps, dealing with regulatory risks, and also AI deployment. And currently this is happening throughout the course of this year and next year, and we're deploying AIDE at 10 NHS trusts within London and the southeast of the UK as well, before potentially going further afield in the UK. And I've mentioned these are some of the hospitals that we're working with, and we're also working with university and industry partners on this as well. So this is the hardware stack that each of those hospital trusts is getting. And this is really great because what this really helps with from a developer point of view is that you've got a standardized hardware and software target to aim for. So at the heart of this is a DGX A100, which is going to be doing the bulk of the processing of all of these different apps and also managing and queuing all of these apps as well. And the other benefit here is that from a patient perspective, all of the patient data remains on the hospital site. So that's really important from a governance point of view as well. And then at the heart of AIDE is MONAI Deploy. So I'm going to talk a bit about how this is implemented in AIDE, but MONAI Deploy is all about packaging, testing, deploying, and running different AI workflows. And so MONAI Deploy is, as many of you probably well know, an international consortium amongst different institutions all around the world. And so really, we see MONAI Deploy as standardizing how we deploy AI packages. And so what I mean by that is, on the left here, we've got an X-ray from 1895. And 100 years later, the DICOM imaging standard came along and was absolutely transformative for healthcare because it meant that images could be sent around hospitals and processed in a very, very standardized way. And if we fast forward a bit further to 2012, this is an X-ray where it's doing some automatic labeling of the hand. We see MONAI Deploy as an equivalent potentially to the DICOM standard, but MONAI Deploy will become a de facto standard for deploying and implementing different medical AI algorithms. So just to describe how that looks, this is how a typical medical imaging AI app might look in a hospital scenario. So you might have some kind of DICOM input. In this case, these are lung CT images, and you would load that DICOM data into your app. In this case, you might want to package that up with MONAI Deploy map package or a map. And within this, which is effectively a Docker container, I'll talk about this a bit more on the next slide as well. You'll load the DICOM data in, will then perform some kind of AI algorithm, potentially using something like MONAI Core, where you segment the lungs, you perform a classification, and then you write that DICOM output. And that will then get passed back to your doctors, basically. And so with AIDE, we're integrating MONAI Deploy at the heart of AIDE, as I've said. So just to take this schematic again, the data might be sent, for instance, that lung CT data might be sent from PACS to AIDE. And then within AIDE, we've got various steps in this workflow. So we've got the MONAI Informatics Gateway, which takes in those DICOM images. They are then passed to the workflow manager. And so this is going to queue all of the different data that comes into AIDE and also assign when it should be triggered, assign priority to different AI apps. For instance, say if you've got an emergency app that really needs instant priority, that will happen here. And then the application server will actually trigger the individual map that is working on a specific data type. So in the last slide, I showed the map surrounded about the lung CT imaging. So that will trigger the reconstruction process there. We have an optional part of AIDE, which is the clinical review mode, which I'll show in the demo, which gives the clinicians the ability to investigate what has come out of the map. So out of one of these two routes, the data is then passed back to the workflow manager. It then goes back through the information gateway, and then it comes back to the original destination where it was sent from. And so if you can develop your apps to adhere to the MONAI maps, then they will be compliant with AIDE. So without further ado, I'll take you through what AIDE currently looks like. So here we have a mock-up of a hospital patient database, so say a PACS. So this is actually something called Orthenc, but in here we've got a patient, anonymous female, 1956, and she's coming for a brain head CT scan. And so on the left here, we can press the send to your server, i.e. send to AIDE button. And so just to really round that home, you can see here it says AIDE. So we can send those brain head CT data to AIDE. In most cases, AIDE will just decide what to do with that data, process the data, and then return it back to that clinician front end. But in certain cases, you might want to investigate what the AI algorithm is actually doing. Say you're testing a new piece of AI, for instance. You want to give doctors confidence in what they're actually about to be using. So that's where this clinical review mode comes in. So in this case, the CT images have come into the clinical review mode, and you might be able to see it, but there's a very small lesion here in this person's brain. And so in this app, this is a stroke phenotyper app. The app automatically segments the different regions of the brain, and it provides lesion parameters for the different areas of the lesion, and then also parameters around how much of that lesion takes up the grey matter, how much of it takes up the white matter. And then if the clinician is happy with these results, they can press the accept button, and that will get passed back into the normal patient reporting pathways. Or if this is a test and we don't want it to go back into the clinical environment, this can just be rejected, and we can continue testing this app if we need to. So that's the clinical review mode. From a administrative point of view as well, we have a dashboard in the back end so that we can monitor all of the different models that might be being performed at that hospital. So in this case, you can see how many executions are being actually run. We can also look at model status, so we can find which models are actually, and by models I mean applications, which applications are being run currently. We can see which ones are stale as well. So if one of the apps hasn't been used for a long time, that's something that is really important, because we want to know why hasn't it been used. Is it a problem with the app that has occurred, or is it that the doctors don't trust that app, for instance? So we can investigate that app and begin to figure out why they're not using it. Down here as well, you can see live which part of the workflow is actually being run and which ones have been successful. And you can even look at the individual different applications and click on them and find out exactly where along in each of those workflows that application is. So in this case, we're looking at that lesion segmenter, and you can see the DICOM conversion step, a bit of pre-processing, the actual segmentation step, then the phenotyping that's taken place, and then the final report that's been issued. And so I also mentioned that AIDE will have an app store as well. So developers will be able to submit their apps, and eventually they will be displayed on this app store, and this will be available to the different hospital trusts that have AIDE deployed, and they will be able to view the apps in more detail. They'll be able to view the intended use of that app. There will also be more details on all of the system requirements. They have a harmonized hardware platform, so this doesn't matter so much in the first instance, but it will give them an indication of how much performance is required for each of these apps. It will also show whether the apps have got CE marking, FDA approval, or UKCA marking. And then we're also encouraging developers to put as much information about their apps as possible to give the doctors confidence for what they're using. And so if they're happy with that app, they can deploy it and liaise with their local trust teams to start using this app. And so in the first wave, AIDE has several apps lined up. I've shown you this AI automated stroke CT analysis app. I've also hinted at the 3D fetal MRI, which I'm personally working on putting into the AIDE App Store. We also have other AI automated brain analysis with the Brain Miner app. We're working with industry on an AI head CT app, and then there's also MR spectroscopy, and there's many more planned for the future. So yeah, that's it for AIDE. Thanks very much for listening, and happy to take any questions.
Great, thanks, Tom. I'm looking to see if we have any questions in Zoom or Discord. Let's see. I don't think I see anything right now, but I did want to mention briefly, I know people had, in our registration survey, we asked about what people wanted to learn about. One of them was also federated learning. And I know you didn't touch on it today, but you guys also have the Flip platform, which I think we are planning to have another MONAI sort of developer day later on in the year that I think we're going to have. I don't know if it would also be you, Tom, or someone else from in the project to talk about Flip a little bit more, but it's something that you guys are using as far as federated learning as well at the NHS, and a platform that you guys are also developing similar to AIDE.
Yeah, exactly. So Flip is actually a partner project developed by the AI Centre along with AIDE, and many of these trusts that I mentioned are also getting the Flip platform, which is the Federated Learning Interoperability Platform. And the idea is that Flip will be connected to the different hospital databases. This is quite a broad overview that I'm giving, but there'll be more information in the future one, but it will be connected to the different hospital databases, and it will be able to perform research studies across multi-institutions data sets, if that makes sense. So and again, that'll be really valuable for doing large-scale medical AI application.
Great. We did have one question come in on Zoom here. So someone asked, can you please go to the deployment app and take us through the same workflow with digital pathology slides instead of x-rays? That would be great. Is that something you'd be able to showcase?
So at the moment, AIDE is working with DICOMs. I don't know if the digital pathology slides are TIFFs or JPEGs or something like this. So there will be some input-output changes that need to be made, but in theory it could work as well. If you can package your apps in a map, then it could potentially work. I know that there's a separate project in the UK which is a bit like AIDE, which is also focusing on digital pathology as well.
Okay. We have another one here. While PACS are supported, are imaging databases like XNAT supported?
Yes. So this is all designed to be open source as well. So again, the AIDE platform, I'm just using PACS as a demonstrator of one for the hospital scenario, basically. So that's very commonly used across hospitals. But if you have an XNAT system, and in fact we do have one, whether it's hooked up to our AIDE system yet, I'm not entirely sure, but you will be able to send DICOMs from AIDE and then sorry, you'll be able to send DICOMs from XNAT to AIDE and then receive them back into an XNAT system. Yeah.
Great. Let's see. It's the
sorry, Michael.
AIDE-PACS. Someone said it's AIDE-PACS VNA. I don't know if that.
What is VNA?
I don't know in this case. So if you could expand on that question, that would be great. Let's see.
One thing as well, whilst we're waiting for an answer on that, Orthanc, I think, is also open source. So you could, in my demo, we could replace Orthanc with XNAT effectively, and it would be the same process.
Yeah. Let's see. Vendor neutral app. So it's AIDE-PACS vendor neutral app. That's what's for VNA.
Yes. So again, PACS is just it's a massive hospital system that I think Sectra is one of the largest PACS providers that we have in the UK. So it effectively has all the different types of imaging data in it, CT, MRI, etc., etc. So yeah, it's vendor independent in that regard. Great.
Let's see. There's a few other ones here that I think, let's see. There are people asking for the slides. So if you get a chance, if you share them in Discord, and then I'll also try to send out slides for all the presenters in sort of a post-event email. It might not be at the end of this week, but it might be at the beginning of next week, where we get the recording up on YouTube, slides, everything else from all the presenters. So we'll add them in Discord now, but look out for them in your email as well as a link for them as well. So let's see, one last question here. Great job, thank you for the presentation. I would like to know what you need to know or to have in order to create an app in AIDE. Can you test or try to create online without software? Just generally how to create an AIDE app.
So the exact, because AIDE builds upon MONAI Deploy, there is more to it, but if you can make your app with MONAI Deploy and put it into a map package, then it will be AIDE compliant once AIDE goes live, if that makes sense. That's the simplest way of doing it. There will be separate documentation for deploying onto AIDE, but effectively the majority of it will be, if you can put it in a map package, it will work.
Great. I think those were everything. If you have any other questions, Tom is in Discord as well. And so definitely mention them in there, and hopefully, Tom, you can stick around for a little bit as well and see if, I think there's at least another one that came in. So again, thank you again for the presentation. That was great.
Thanks. Thanks, everyone.
Cool. So next up, let me go ahead and take over and share here really quickly.
Yeah, thanks.
So next up, we have MONAI Label Reviewer. So MONAI Label Reviewer was contributed by Charité University Berlin. And so I think we have two presenters. Keno, you're here. Is Minh also going to be presenting today? Should I put him on as a panelist?
Yes, please. We do a split presentation. So I will do a quick introduction and he will do the main part.
Okay, so I'm going to go ahead and promote him to a panelist right now. So go ahead and tell us a little bit more about MONAI Label Reviewer and take it away, introduce yourselves and your work you guys are doing.
Yes. So before we start with MONAI Label Reviewer, I want to introduce the working group real quick and some experience with MONAI Label. So we are an interdisciplinary working group at Charité Hospital in Berlin, and our main focus lies on AI for radiology. So we develop applications for chest X-ray, chest CT, and chest X-ray is also a use case for MONAI Label Reviewer. Also prostate cancer, working on kidney cancer, a big part is axial spondyloarthritis. So we have a big center on this here. Some on shoulder fractures, and recently also exploring medical NLP focused on German medical texts. So we started using MONAI Label at the beginning of this year. So, there is a use case where we wanted to have the anatomy segmented in chest X-ray, to use this as a basis for other downstream tasks to know where our predictions are in the image. So we started with, I think, IDK Snap to label those images and then switch to Slicer and just load them from a file system. This took about 50 minutes per image to label, to draw contours around the heart and the lungs. We got faster over time, but it was really slow because people had to load open images. Then we switched to MONAI Label. This really speeds up the annotation. So by this time, I also started to log the times it took us to complete each image. And as you can see on the graph, it was roughly 250 seconds. So let's say 4 to 5 minutes per image. Of course, there were outliers. Some images are really, really complicated. We got better over time, as you can see, consolidating here. And then we introduced a new reviewer, and then again, a bit more diverging in the images. So at February, we had quite a lot of images, and we started that it would now be a good time to train our first model, see how it performs, and performed quite well. So we decided to use the UNET backbone from MONAI label. And this really was a speed up. So you can see drastically reduced the times it took us to label the images, and also much more consistent. So a double speed up to label an image. The only problem was the segmentation algorithm was already too good. So not all images are difficult in the data set. And the segmentation algorithm was quite good at segmenting those easy images. So they were very fast. Sometimes it was just loading the image, doing the inference, and that's it, no corrections necessary. So we decided it would be better to only see very, very difficult images. So that's when we started to look into the epistemic scoring of the images, which also really was very helpful. We took our model, and what it does is it activates dropout doing inference. So you run it several times and you get slightly different predictions. And the assumption is that if it's an easy image and the model is sure and is very good, these slightly different predictions will greatly overlap. But if it's a difficult image, those predictions do not overlap. So you can basically calculate the score, and the lowest score is the more difficult the image is, and then you can push that forward to only annotate the difficult images. This worked really, really well. So the first image we got out of the data set is like 20,000, was completely white. So there was a false image we did not know existed. The second image was a patient where the colon collapsed into the lungs on both sides, also very, very difficult image. And still, despite having all those difficult cases, we still were faster with MONAI label than we were at the beginning. So this really, really helped. But there was another problem we wanted to address. And this is that we wanted to have a simulation of a radiology workflow in MONAI label. So I'm a radiologist myself, and I'm now a consultant in radiology. And what happens is you have an exam there, probably you know. So residents write the report. They also help with the exam. And then they finish their report. It's a preliminary report, and the consultant proofreads the report. If there are small mistakes, maybe a small finding was missed or typos, they finalize the report by themselves and send it back to the patients. If there is a large mistake, so significant finding is missed, or it's just not in good shape, like residents were very, very early in their career, they might send back the report to the resident to request changes. And consultants are much more expensive to have, and they are very, very busy. So if you do labeling, usually you have residents, and usually they are early in their career, but they have more time then. And that's why I also wanted to have this workflow in MONAI labels, to have a reviewing tool, which Luke will now introduce to you, which allows more experienced readers to correct work of the not so experienced readers, but in a much faster way. So they can go through the images, approve the images, and maybe correct the images if necessary. Right, so I give to you, Luke.
Hello, now you can hear me. I shared my screen so far. Can you see the screen with the MONAI label reviewer so far, just for the check?
Yeah, we can see it.
So that's our tool we created for our purpose to increase the quality assessment of the segmentation. You can find it in the slicer tool below in the tab of active learning below the MONAI label, our MONAI label reviewer. So if you click on that, you can see it starts automatically in the basic mode. And apart from that, we have also a so-called reviewers mode. I will go through with you through the basic mode as first. So you can see here, you have to type in the URL of the MONAI label server. In this case, it is locally on my machine. Click on connect and it will load the entire data set. And you can see here the progress bar, which represents the number of images which has segmented already. So in this case, 494 of 1000. And now you can see here the previous and next button, which allows the reviewer to browse through the data set. I click on that one, for example, and will load automatically the next one. And also you can see here a horizontal slider, which shows the reviewer at which image he is currently looking at on the right box. And another point which we implemented is the version control. So the reviewer can select between different versions for segmentation. For example, if the reviewer wants to improve it and save it, I will come to that later because that function will be activated in the reviewers mode. What you can see below is the editor tool. Most of you who have already used the MONAI label and the slicer knows this toolbox. You can click on the eye here and hide and show the corresponding layers to improve the segmentation. Correct. And next are these metadata like the image ID, the annotator, and the annotation state. Right. And what you can also do as a reviewer is to select a subset of images. So you can see here a combo box where you can choose one annotator, for example, annotator _2. Click on that and will automatically just load the images of annotator _2. In this case, 389 of 1000. Yeah, that's it so far for the basic mode. What comes more is now the reviewers mode. I click on that, you can see here. And then you have to type in the reviewer's name. I type in just my name and click on connect. And you can see here already there are more, just the previous and next button. What is additionally, you can see here the easy, medium, hard button, which allows the reviewer and the annotator to classify the images according to the difficulty level of segmentation. So in our case, we ask for the most experienced radiologists to look through all the images which are very hard or just medium, and significantly improve our data set. What comes more is the flag and approve button. If you click on that, you can see automatically it will change here on approved, and it allows now the image to be part of a data set to train a machine learning or deep learning model. Flag allows our team to pre-select such data sets which are very complicated, which then should be overviewed by very experienced radiologists as well. And we can type in here some comments on that just for further information. Correct. So additionally, what we just implemented recently is now that the reviewer can instantly edit the segmentation or improve it. So just click on that button. And now you can see on the left side here all the tools which you can use. So I demonstrate that once. I click on this tool and just put here an X for demonstration purposes. And then the reviewer can choose between 3 options. The first one is to override the current version, save as a new version, or delete the version. We can see here now final and version _1. And now I click save as a new version, confirm this. And you can see it takes some time. I'm sorry for that. There's a small problem. I will solve it later. But actually what should happen is that it appears here version _2. Correct. So that gave us the very experienced radiologist to improve it instantly. Now I come to next. I just want to reload it once again, show it view and type in my name again. There are new functions as well, which comes with search images. So I close that and expand this section. And here the reviewer can type in the IDs of the images, which are comma separated. And I just choose that one just to type in and then click on search. and it will appear on the right side the list of all images which has been found, indicated by the second column, yes, found, and no, the dummy wasn't found. And this third column represents if it's already segmented or not. And you can click on this, for example, then click on show, and then you can see an image without segmentation, or the first one which has been already loaded previously. This image with segmentation. Furthermore, you can pre-select the annotators. So for example, annotator _2 and all the flag images. And click on search. You can see here, these were the 4 images which has been flagged by annotator _2. Yes, and the last functionality is the quality. So you can just select hard for all the images which were hard to segment it. Click on that and you will see probably note, there are 5 images, for example. Yes, that are the main functionalities of our tool. And what I haven't shown so far is that one. Exactly, if you initially just want to see the images of annotators _2, browse through it, it will load the entire data set. And now you can pre-select the filters again here, just like the approved ones or the flagged ones. And then you have to click on load again. And you see again in the horizontal slider, here are 4 images which were flagged by the segmentator. Right, that's it for the presentation of the tool. I hope very much it will be used a lot and it's beneficial for each research group. And there are some improvements and limitations which I would like to explain for short. I'm actually a developer coming from the Java world. And for me, it's very important that the entire data can be stored in, let's say, SQL or NoSQL database for the purpose of a big data set, big data. So we can store more metadata in a large scale. The second, what's important for me as well is that we can just request very complex metadata by standard SQL syntax. For now, I implement a lot of code just to realize all these filter options. And what comes more with a SQL database would be that all other services and software could access to the entire data. And what is important for me, lastly, is the readability of code. So I use consequently the library typing just for indicating what type of variables we are using. So that's actually helpful to look through the code very quickly. And what should be implemented is really concepts of clean code. That's, from my side. Thank you very much.
Great, thank you two for that presentation. We have at least 2 questions here that I'll kind of answer. One is a bit more of a statement, but we can ask always to expand on it. So let me go ahead and share my screen. I have them because one is from Zoom and one is in Discord. So I'll go ahead and share here. So number 33 here was, this is one of the best recent contributions for MONAI. They're working on this as well. And they said that they still have some issues, especially with time to process. I know that just generally, and I'll cover this after this for the next presentation for some of the roadmap pieces. For MONAI Label itself, we're working on performance on that. And it's one of the big things just generally with MONAI Label and sort of the process in between the viewer and label itself. I don't know if you have any other issues or specific things on time to process with what you meant specifically there. But if you do have any other comments you want to make, we can always ask them in Discord and we can maybe try to address any of them. Or you could definitely go through to the GitHub page and put an issue in there if you're seeing something very specific with one particular piece of it that you're seeing issues with in general processing or anything else. That way we know, again, that way we can kind of address that or know that that's sort of an issue in there. But I'll talk a little bit more about some of the roadmap and things for MONAI Label. Keno or Do you have anything else that you want to mention generally for sort of the time to process or sort of the workflow right now or anything that's being worked on?
So we found out that you have to frequently clear the slicer cache if you work with slicer. It just fills up and it takes longer and longer and longer to load the images. The beginning is very fast, but after 500 images, it was 1 minute loading time. and just clearing the slicer cache will solve that issue. And what will not solve the issue is to limit the slicer cache. It will just continue writing the cache, although you say 20 megabyte cache, it will just continue caching files. You have to manually delete the cache. That was our workaround. And if you do that frequently, then you speed up the loading times, annotation times.
Okay. And someone said, yeah, sure, they'll put an issue with the timeframes as well. So it looks like they're going to submit something to GitHub. And again, anyone else who is, I know there's been conversations about different things, not just for this presentation, but for other ones before that I see in Discord, a lot of conversations going on there. Always feel free to go to GitHub, and we have, let me see. Right here was actually where MONAI Label Reviewer is for here in MONAI Label, but for anything that we presented today, go through and submit an issue. You can always go through, put an issue in here if there's one specific issue, or start a discussion about what you're seeing, timeframes, timelines, support for different things, whatever you're interested in. Definitely go through and contribute back here so that we know it's an issue, or again, just general discussion of things you'd like to see within any of these things we've been presenting. So next question was, this demonstration looks great. I was wondering if it was also possible to process contour annotations rather than voxel masks showed here in this way.
So we have so far just used it on thorax X-rays and CT scans, and just used a proper editor tool which was already provided from the slicer. So we didn't try it out, the contour annotation so far, I guess.
Okay.
Yeah. I think it can be extended if it's missing.
Okay. And then someone, they asked about cleaning the cache for slicer, or what cache specifically, and that was the slicer cache, just going through and manually deleting it. Are there instructions anywhere for that you guys might be able to link or mention in either Discord or anything on the slicer docs that might be able to help make sure people know how to actually properly clear the cache for that?
Yeah, we can do. It was very easy, just 1 or 2 clicks. I will look it up and write it to Discord. Okay,
great. And then one more for MONAI label development, and this, you guys can definitely talk to this, is does one require background for slicer development to be able to kind of work with MONAI labels? I think generally MONAI label, no, but you guys did something in 3D Slicer. What was sort of your background before in doing plugins or things for Slicer? What was sort of that that you had before creating the reviewer?
Yes, so in my case, I am a computer scientist in my master's program, so it was quite easy for me to look through it. But developing a slicer extension is not that easy because all the code is very extensively just in one file. So I can recommend you the book Clean Code. It's very instructive, which give you a feeling how code should be written that other contributors, other developers can read it very fast and quickly. So it takes me approximately 1 or 2 months to understand the entire code. Yeah.
Okay, great. So I put that book recommendation there. There's actually in one of our, and we're going to go into a little bit more into one of our videos, but for MONAI label, generally we're putting out video series right now, and one of them touches on some of sort of the Qt extension viewer, and I think we're going to go more into that in a future video. It might be a little while till we are able to get around to it, but it's pretty easy to go through and look at some of the slicer extension pieces from it. But a recommendation of just generally how to lay out your code in a way that's accessible to other people is definitely helpful. And generally for MONAI label, it really depends on which piece you want to work on. If you are wanting to contribute back maybe a sample application with whatever you're working on, I mean, if you already are doing deep learning, and you want to implement a particular network or sample app that's there, or a model that you ended up developing that was an annotation model, you can contribute that back to Model Zoo. There's a lot of ways just generally with MONAI that you can contribute. So if something doesn't fit necessarily your background, you still might be able to contribute back. And if you are kind of struggling or need some guidance, always reach out. I don't know if Andres was able to jump on yet, but Andres is also one of the co-creators of MONAI Label. He's always very active. He does Project Week demonstrations. Just there was a Project Week back in June that he was on showing MONAI Label, helping with projects around there. And so there's a lot of good people who are kind of mentors like to the community who are willing to help you sort of go through that development process and figure something out. But the only way we'll kind of know that is if you are on Slack or on GitHub and asking for help and reaching out to the community for some of that help and guidance. I don't see any other questions. So thank you both for the presentation.
Another comment?
Oh yeah, go ahead.
For the contribution, I would also want to ask the community for contribution to the revenue tool, as our funding, which we used to spend a lot of time to developing the tool, is now running out. So we won't be able to push that much further, and would be very happy if we could give it back to the community. We still try to add some more features, but will not be as frequent, and we can't get that involved as we have been last half year. So it would be really great if there are any contributions to add functionalities, because we hope that it will really speed up annotations and increase label quality for everyone. So thank you very much.
Great. Thank you for that. Yeah, if you have any other questions, they're both in Discord, so feel free to ask anything else there. And thank you too for the presentation and demo. That's great. And thank you for the contribution. I think a lot of people have been using it, and it's been great having such a big contribution to MONAI and MONAI Label. So thank you too again. Okay, I know we're getting close to the end of the meetup, and we still have at least one other section here that I was going to go over very quickly. That's sort of the roadmap, which I can go over relatively quickly in probably like 5 minutes or so. And then sort of the office hours. The office hours were kind of intended to be a little bit more open. People are talking in Discord. We have some voice channels set up in there in Discord that you can jump into, chat with people if you want to chat in there after the Zoom meeting is over. So I encourage you to do that. I'll be around on Discord for a little while longer after the event. I am in Pacific time zone, so it's about to hit 2 o'clock in the morning for me. So I probably won't be up too much longer, but I know a lot of other people are in Europe time zone or sort of Asia, East Asia time zone. So they'll probably be up a little longer and around a little longer than I will be, but I'll check tomorrow as well for anyone who has questions. Again, feel free to reach out on Discord, Slack, LinkedIn, whatever makes sense for you. And I can always help connect you or get you some answers if you need anything. But I'm going to jump to roadmap really quickly, try to go over it in the next few minutes here. So for MONAI Roadmap, really this is talking about, well, what's next for MONAI? So before I do that, I'm going to talk about our latest release. So we just had releases here in June and July around MONAI Label, Deploy, and Core. And I'm going to touch very briefly on some of those things, which you've already heard about some of them throughout here. So I won't go over it in a lot of detail, but MONAI Label, pathology sample applications were released, segmentation version, a deep edit version, and a new click version. And some of the speed up for inferences were around, for nuclei segmentation, we're up to about 6x speed up, anywhere from about a 20-minute inference down to a 2 to 3 minute inference, depending on the size of the image and the number of nuclei that were there. We have a video a little bit that shows some of the numbers on YouTube around MONAI for pathology. And we're working on some other videos next up as well for the digital slide archive integration and QuPath integration to show you how to get started with that. Viewer integration, DSA, QuPath, CVAT are some of the integrations that we recently added for things. And then some update to the Scribbles annotation methods that if you're trying to use Scribbles annotation, which we also have a video for that up on YouTube, there are some updates to there. Then there's a drop down now to be able to pick the type of method that you're using for that. MONAI Core, Eric gave a great recap of MONAI bundles, what they are, going in depth to everything about MONAI bundles. So those were recently added. Different transformer networks, we had SWIN UNETR and Ahmad who got to talk about that. Deep Edit and NuClick were added in there as a basis for some of the pathology apps that are MONAI Label now. And then there's some additional things around 2D and 3D bounding boxes, Retina architectures, and some general box utility functions that were added in there. So those are things that are going to be worked on more in the future, but sort of, I think, the basis for some of those things that were added in for this latest release. And then MONAI Deploy, the MONAI bundle inference operator. So if you're creating an AI application, it's pretty easy to get started with the operators. And if you're using a MONAI bundle, there is an operator that will load that, and it'll do both in-memory and file IO loading, and really easy to get started. There are great tutorials on the MONAI documentation or MONAI Deploy documentation for getting started on general operators. And then also for the bundle operator, there is a new, I think, 6th or 7th notebook in there that talks about how to utilize that bundle operator in your map. Multimodal support. So there are bundle operators are a single model, but you can have maps can have multiple bundles. And so general support for multimodal support. And then there's some additional things around informatics gateway and workflow manager that were added in as well recently. So I encourage you to go check out the documentation for that. And Thomas touched a little bit on that general workflow for deploy of map to informatics gateway to workflow manager to whatever other system, Argo-based system, or whatever else you might have connected to it in sort of your deployment workflow. So for our roadmap for all the different products, we have for MONAI Label, we have a rough target of Makai for a release, and that one's going to be kickstarting a little bit more around the Kai part of Makai. So things around endoscopy. So there's going to be general kickstart of how do you do annotation for video, and CVAT is one of the ones I think that they're working on to figure out what that goes into. And then general active learning. So there's been some discussion around active learning. What does that look like? What are we doing in it? And it's a active area of research for us because it really differs per type of imaging that we're doing. For something like endoscopy, you can think of the images being, since it's a video, it's a little bit different of how you might pick out that image versus something like MR or CT versus pathology. So just general work into active learning, and then more performance optimization. So we're working on sort of debugging what that process looks like as far as there are copies or excessive copies in certain areas that are slowing things down, even by a second or two, that are being worked on. And so just general performance optimizations. For RSNA timeframe, end of the year, a little bit more MONAI Label integrated solution for radiologists, including things like XNAT, reviewer dashboard for radiologists, multi-user deployment and scalability, and secure server infrastructure. So a lot more around sort of multi-node deployment and a little bit more integrated solution work there. And then later on, early next year, again, just general more work around active learning, some experiment management and versioning support for things, and then starting to look at mainstream commercial integration. So what does it look like to actually get integration into some of the actual solutions that are out there to make it even more seamless to be able to use something like MONAI Label in some of those commercial products that are already out there. For MONAI Core, we talked a little bit about the MONAI Model Zoo. There's that survey. I'll link it again afterward as well. I encourage you to take the surveys to look at the discussions that are out there already. Contributions to the Model Zoo. If you fill out that survey, we'll reach out to you. If you're interested in getting a model that you're working on into the Model Zoo as a MONAI bundle. But we also have things around AutoML and creating a more AutoML sort of turnkey solution, making it really easy for you to get started with AutoML-based solutions for things. And then, again, things around Kai. So what does it look like to develop things around like endoscopy? And then we'll also talk a little bit more about federated learning and integration of federated learning. So we have some examples right now of NVIDIA Flare, Substrata, a few other federated learning tools in the MONAI tutorials repo right now. So if you go to the tutorials repo, federated learning folder, I think there's 3 examples in there. But we're having a MONAI federated learning spec, and the first framework that's going to adapt to that is going to be NVIDIA Flare. And so we'll be showcasing some of that as we get closer to Makai. For RSNA, more enterprise style infrastructure solutions. So working with a lot of the CSPs, GCP, AWS, NVIDIA to figure out what does that look like to do that at a little bit more enterprise scale, and having a nicer turnkey solution for AutoML, and some research modules into reconstruction. And then last, early next year at around spring timeframe, a little bit more around software framework to compare and develop state of the art. So what this means is we have a lot of methods for you to get started, whether that's DINST, whether that's UNETR, whether that's SWIN UNETR, or some of the other networks that are in there. What does it look like to have an easy way to compare between these different methods if you're just getting started in research and what you're actually doing? And a way to kind of, how do you compare these different solutions and what works best for you? So some work around what does that look like, some guidance for people who are getting started and being able to do some of this state-of-the-art research and applying it to your own models and own tasks that you're working on. And then more around things like multimodal workflows. And then I think somewhere in here, and I'll touch about it a little bit more on another slide, is Eric talked about ECG and other things that are being worked on and being introduced as well. So I'll show you the links for those here in just a moment. And then for MONAI Deploy, generally more work around Informatics Gateway and Workflow Manager and an Argo-based pipeline orchestration example around McKay timeframe. So that's really fleshing out more of the story that was talked about for the AIDE platform and what that really looks like. And then there's going to be deployments. So like Thomas talked about, deployments are going to be at 10 different trusts and really trying to showcase what that workflow looks like. And then last, going into next year, some more MVPs around what does that reference architecture really look like? And what does that look like for you to sort of take that and have this architecture ready to go and where you can kind of plug in your pieces to get it stood up for whatever you're working on. And then more integration into general healthcare IT platforms. So looking at what integration pieces that might be needed and seeing what that integration looks like to other general IT platforms. So how can you get involved in MONAI? I've mentioned it and other people have mentioned it again and again throughout this meetup, but there's a bunch of different ways you can get involved. So whatever makes sense for you. So maybe that's just starting to use MONAI. I know a lot of people here have never used MONAI as part of that registration. I think about 40 to 50% haven't used it. So a good way to start is just go and use it. There are tutorials in our tutorials repo, a lot of different ones there. They have Google Colab links that you can click on, go to Google Colab and it'll launch Google Colab and you can easily get started that way, or pull it down, clone it, and work through them on your own machine or workstation. Submit issues or bugs. So if you run into issues or bugs or anything else, submit them on there. Let us know that there's an issue, that you're having an issue, and we'll try to help you through it and figure out a fix for it. And then last, contribution of code, features, research, or just even discussions or models. So code contribution, MONAI Label Reviewer is a good example of that, of a great effort being contributed to MONAI itself in the repo. Different features or research, so there's been a lot of things around SWIN UNETR of different things that could be implemented and showcased that were implemented at some point, but maybe not contributed back to the repo. So getting those back into the repo so people can see and look at the research that's being done, or different things that are being helpful to you and your workflow are probably helpful to other people. So it's a great place to contribute back. And if you need help, reach out to one of us, start a discussion, and we're always more than willing to help you get started on what that looks like. There are a few surveys and discussions around that are active right now. MONAI Model Zoo survey I mentioned. There's a pathology survey that you can go through. And if you're in pathology, you want to give some guidance to what the working group can kind of look into as far as next things to implement, I encourage you to go do that. There's the ECG discussion that is actively going right now, and some contribution of code to MONAI core itself. And then there's a biomedical applications feature proposal and discussion as well that are actively happening now. So another good one to kind of go take a look at for just general biomedical application happening. Last, we have a lot of resources. I'm not going to go over them all here, but just to kind of highlight, we have Slack, YouTube, Medium, GitHub, Twitter, our website, everything you can kind of go through and look through all these. They're on the MONAI website somewhere or another, but we'll share these slides and you can have access to all of these resources. And I'll also post some of them in Discord as well for you, so you can easily go through and find all of them. But lots of ways to get started. We're actively working on more YouTube videos. So if you have something that you're like, hey, this would be great to have something that it was difficult to get started using whatever feature or whatever thing, let us know, because we're trying to aggregate a list of things like videos or documentation updates that we can start integrating into all of our MONAI sort of onboarding developer experience pieces. So with that said, that's sort of the end of the official meetup. We'll have sort of this office hours general Q&A. I'll go ahead and stop the recording, but I'll see if there's any other questions, and whoever else is around who's part of the MONAI core developer team or people who presented, I'll see if there's any other questions from there. But for those of you who need to take off, thank you for joining. Again, reach out to any of us who were on here. I'll be in Discord tomorrow as well, checking in there. And then we'll head over to our office hours right after this. So I'll end the recording. And again, thank you all for joining.